That was a lot. So if you're sitting in the audience like I am, I'm thinking, boy, how am I going to put all that into what we're doing today? But it was really an amazing overview. And I think I'll just say two things that Mike sort of said. Thank you all for being here. Congratulations on the work you're doing. Today you're going to get to see hopefully what will be like a shot of adrenaline or coffee to see all the different things people are doing so we can hopefully come to a better place. And there's no better place to start, I think, than think about how do we take that overview talk of precision to personalization to actually have a person with one of those conditions come up and talk to us about that. So I'll have Katie Wright, who's, as you heard, a podcaster, but has been living with VEDS, and she's going to come up as a person who's been living with it and talking about it and share her story and some of the things she's been doing. As I said, we want questions. So after her talk, I know there's a lot of talks, but I'm going to ask questions for her. And if you have an immense question right away, please make sure you get it so you can try to answer those. So I'll have Katie come up, and she's a patient advocate, as you've heard, podcaster for vascular Ehlers-Danlos syndrome. Thanks.
Yeah, thank you. I'll just take this and move it down. Hi, everybody. So, as mentioned, my name is Katie Wright. I have vascular Ehlers-Danlos syndrome, or VEDS, and I was diagnosed in 2017 at the age of 28 years old with this condition that I was born with. I'm very grateful to be here. I'm going to start by talking to you a little bit about my diagnosis story because I think it's really relevant to the talk that we're having today. So in 2016, I was in my mid-20s, had no idea that I had vascular EDS. I'm just going to call it VEDS from here on out. I woke up with what I described as the worst neck pain I had ever experienced in my life. It was in March of 2016. I described it as if my head was going to fall off. I tried to make it to work. I made it maybe about an hour, and then I left and went to my PCP, who diagnosed me with a pulled muscle or a sprained muscle, gave me an anti-inflammatory and a muscle relaxer, and sent me on my way. And I think it was a couple weeks that I, made it with all of those muscle relaxers, ran out of them, started living on ibuprofen and Tylenol for the next couple months. And I really got into this pattern of, for a couple months, I'd have this really intense pain like this. It would finally start to get more livable, and then it just seemed to me as a patient, the day that I felt totally better is when it would come back very spontaneously, felt like this really tearing pain. The first time that it recurred, I was reaching down to get a blanket off the floor that my cat had very lovingly dragged onto the floor, and I picked it up, and then all of a sudden, it was this severe tearing pain, and I didn't know what was going on. I didn't know that I had a risk of dissections and artery things and all these scary things that I now know that I'm subject to. So this went on for about a year with this pattern of recurring, spontaneous, severe pain with no answer. I visited a chiropractor. I did a lot of massage. I was doing some really intense stretches that I now know I probably should not have been doing. And you're all in vascular here, so you probably know I had a carotid artery dissection and didn't know it. But I still didn't know this about a year in, and I was going to be worked up at a vascular surgeon's office for potentially thoracic outlet syndrome. They were really trying to figure out why was this pain here. Maybe it had something to do with the reaching, because the third time it happened, I was reaching for a shampoo bottle in the shower. So we were just kind of fixated on the wrong thing. And I was at lunch with my brother, and he made this joke. They won't even have to do a scan because your skin is so see-through that you can see all the veins in your body. And we had a really good laugh about that because that was true. It's been like that my entire life. But I went home after lunch that day, and I just knew that somebody had missed something along the way. And my whole life I had felt like there was some mystery that if I just had a doctor house, that he would come in and figure it out. So I sat there in my living room that night. I pulled out my medical box of medical records. I had had some weird things in my life happen, and I pulled it out, and I was like, okay, I'm going to figure this out. I wrote down things about myself that had always been there, like the visible veins, like my hands. I'm slight acrogyria, and that, developed when I was in elementary school. So that was one of the things throughout my life where a doctor had said, oh, you have scleroderma, so because of your hands, and had medical students come in. But it was never scleroderma. So I wrote that down, had an ACL tear when I was 18 from a very simple fall, tore my ACL almost all the way through, things like that. And then I found this biopsy report from something I had called elastosis perforans serpaginosa, which is a rare skin disorder where your body kind of pushes the elastin tissue out of the skin in a serpinaceous or snake-like pattern. And on that biopsy report was a note from the dermatologist that sometimes this is associated with a rare condition called Ehlers-Danlos syndrome. And then all this information came flooding back to me, because at the time that I saw that dermatologist, I was a full-time college student with 12 to 15 credit class load and worked 40 hours a week. And not a lot outside of that got stuck in my brain. It was just something happened, handled it, moved on with my life, gone forever. But I remembered all of this stuff came flooding back, and I had seen a geneticist in 2014 for this possible Ehlers-Danlos syndrome thing that does not run in my family. And that dermatologist, he sent me to the geneticist. I had the genetics appointment. I had briefly looked at vascular Ehlers-Danlos syndrome, and I asked the geneticist about it, and I was reassured I don't meet any of the criteria for Ehlers-Danlos syndrome, especially not the vascular type. And I moved on with my life. So here I am in 2017 now, and I'm looking at the NIH website on vascular Ehlers-Danlos syndrome, and I am absolutely devastated because it comes with a shortened life expectancy and a lot of really scary things, like artery dissections and aortic dissections, bowel perforations, lung collapse, uterine rupture. The list is very, very scary when you look at it without any medical guidance in your living room. So that started my journey to diagnosis, really. 3 months later, after a lot of pushing, I got a genetic test and confirmed that I do have VEDS. And 4 months after that, I finally got to see Dr. Shireen Shalhoub at the University of Washington to take a look at my baseline scans. And we found this internal carotid artery dissection with a little pseudoaneurysm in my left internal carotid artery that explained the pain I was in. Pretty much as soon as I got the diagnosis of VEDS, I was told, maybe stop all that intense stretching until we know what's causing that neck pain, because, could be a dissection. And that kind of led me on the right path. I think that since the diagnosis of VEDS, I got really involved in the community. Shireen Shalhoub invited me to join what she was starting, the VEDS Collaborative. And that was a research project really designed to bring patients into the discussion with researchers and guide research in a patient-oriented direction. And I got really, really passionate about that. I joined her in 2018 on the advisory board. I started a podcast where I talked to people about their stories with VEDS and now similar conditions like Marfan syndrome and Loeys-Dietz. And that really kick-started my involvement in this wonderful growing community of people who need answers. Medically, since my diagnosis, I've had several mini strokes over the course of a year. And in 2021, my spleen ruptured spontaneously when I was getting a soda out of my fridge. And that scenario for me was really important when I think about precision medicine, because at that time in 2021, I knew I had VEDS. I knew the risk factors, like the risk for the types of things that could happen to me. I knew what to look out for. And when I had that sudden, spontaneous pain, I knew instantly I was either internally bleeding or I was having an aortic dissection. So I immediately called 911, let them know where I was, and I told them those words. I'm sure they don't get that very often, but that's what they got. And then I called my vascular surgeon, Dr. Shireen Shalhoub. And I said, hey, I'm trying not to panic. I think I'm bleeding internally. I was laying on my couch, feet up, so all my blood would stay at my head and I wouldn't pass out. And it just she just kicked into gear. She called the hospital, let them know I was coming. She had her resident there at the ER, ready for when I got there. And then she called me back and stayed on the phone with me until the first responders arrived so that she could tell them, I needed to go to this hospital. They were waiting for me. I have this. It's serious. Let's get going. And I think about that a lot, about what the situation would have been like if I didn't know that I had that risk, if I didn't have a vascular surgeon on my team kind of helping guide my care with vascular EDS. And I still think about the things that could have been better. When we got to the ER, the resident, he continually pushed for me to get that CTA. It took, I think, 2.5 to 3 hours still to get that CTA to confirm that I had 3 units of blood in my abdomen and I needed to be admitted. And that was with knowing the diagnosis, knowing I could be internally bleeding, having a resident there on the floor who was just constantly, I think I saw that guy every half hour. He was amazing, just continually pushing. We're going to see what's going on with that. And that could be better. That wait time could have been improved, and it could have not had such a good outcome. I might not be standing here today. So that's what I think about when I think about precision medicine. I think about knowing the risks that come along with my condition, and I think about how to detect these emergencies faster. I think about how to diagnose conditions like these faster. What if in 2016, 2014, when I saw that geneticist, there was better guidelines on who should get that genetic testing to check. And I think a lot of what I'm really excited about today is the AI stuff here on diseases and recognizing them. And I think there's a whole world of possibility with emergencies and recognition of disease. I think about how to prevent these scary things that can happen to me and to people with similar conditions like mine, like Marfan and Loeys Dietz. And I really think about the personalized care that somebody with my condition needs. I think it's really important, and it becomes very difficult when you run into providers who don't necessarily understand that need for personalized medicine. I think about how we need a treatment to prevent these scary things from happening, and it is very precise. We know the genes that cause these conditions, and there's a potential there for a way to fix them. And I think about how to prevent the devastating losses in our community that happen both to people who know they have the diagnosis and run into challenges in the ER, or we just don't have a treatment, things go wrong. I really think about how to prevent those losses a lot. I think to me, a focus on precision medicine and genetic predisposition to risk combined with the identification of biomarkers to detect these kinds of things that can happen and a treatment are really critical to progress. And tools to recognize and diagnose these conditions and the emergencies that come with them are critical, as well as methods to impact care in the ER. And we have a growing community of people who are really interested in research for this, and I don't see any reason why it's not possible. We have a track record of a very engaged community that wants change. They want to be involved in research. And when we're talking about, looking at all the data, I think you have a lot of people out there who would love to see change in research. So I don't see any reason why it's not possible. That's it. Thank you.
I'll just
Sure.
start by saying thank you again.
Yeah,
really
thank you.
sort of centers us. We're talking about precision. We're talking about personalization. What a powerful story. So thank you again, because I think it's going to help us every day come back to sort of the North Star of what are we trying to accomplish. I see one question in the audience.
Sure.
You want to? I think there's a microphone, or if you want to, I think we're small enough where you could just.
Yeah, I'll just speak up.
Yeah.
That's a very moving story.
Thank you. Thank you.
I got this right here. You can. Yeah. Do mine. Get some steps.
So we heard Dr. Damrauer's talk this morning, which was really comprehensive, really interesting, fascinating, and we also learned that, practically speaking, when we do these genome-wide studies to diagnose conditions, in his case, he was looking at coronary artery disease, or this is the example I want to bring up. It was most useful in younger, or more useful in younger patients. So I'm curious, from your perspective, should we all be getting these genome-wide tests at birth and be burdened on the one hand with all this information of what could happen? Or, how would you approach that from your perspective?
Yeah, I mean, from a patient perspective, that's a super interesting question. My first instinct is, yeah, definitely. But there are a lot of things that come along with that, as you mentioned, knowing the risk, not having a treatment. What do you do with that information? How does it guide your life? Another challenge, is in our current system, if you are diagnosed with this condition, you're not eligible for life insurance or disability anymore. And so then are you now predisposing a baby who had no choice in the matter to be diagnosed to never being able to qualify for life insurance? Is that a reason not to diagnose? I don't think so. I think it's a reason to adjust the other system. There are things that we do with these conditions. People who know younger, they can adapt their lifestyle to avoid high-risk activities, right? They maybe don't play football or really risky collision sports. They don't go into really, really heavy bodybuilding or anything like that. So when you're a child and you grow up knowing, I think there's a lot of potential benefit for how you can handle that information. Generally, the experts have us get scans on a regular basis too to monitor for new aneurysms and dissections, because these things can happen out of the blue, like my spleen ruptured when I was getting a soda out of the fridge. These things can just happen. And when you're monitoring these things, you can go in and do planned procedures rather than waiting for them to become emergencies where you're in that emergency room, perhaps bleeding out, and there's no choice in how you address that issue. So to answer your question, I think I lean towards, if we were able to get newborn testing for these very specific genetic diagnoses, I would be on board with that. But I would think it would require an adjustment in how we handle life insurance and disability on a larger scale.
It's a really powerful question, and there's going to be a variety of conversations in the panel too, because there's all this stuff for certain newborn conditions versus others, and obviously what can we change and how do we do it. But the promise that you told us about of the story, because when you sit in the audience and you hear all the little markers, in retrospect, everything's easier to understand, but you would love a system that was taking that data and saying, you're getting closer and closer to potentially a set of diagnoses. There's one more question, and then yeah?
Hi, thank you so much for sharing your story. And I'm very excited that you are excited about new technology, especially AI. My question's a little different, and it's a little bit related to what Dr. Alami just said. And he talked about, and you addressed the pragmatic issues of having too much information and how that affects healthcare delivery for certain patients. Patients, and in this case for genetic stuff, I think that makes sense. I think my question really is, patients seems to be very divided on how we move forward with AI, for example. And Dr. Dermar mentioned some of the black box issues. How do we tackle that conversation with our patients? And to you, I think for you as a patient, what has been the most powerful type of language people have used for you to assuage those fears? Because it's coming. I don't think we can get away from it. But people are very divided and very fearful of new technology.
I mean, I think the biggest thing is to get patients into a room and involve them in the conversation, because everybody's going to have a different answer on that, and as you mentioned, some people like myself are very excited about the potential for it, and other people are very hesitant. But in order to assuage the fears, you have to get people into a room and just have an open discussion, understand what those fears are, and you can't do that without having people as part of the discussion. So I go back to, when I was invited into the VEDS Collaborative and was invited to provide my perspective and be a part of the research itself, and how life-changing that was to be a part of the conversation. That might be a simple answer, right? It takes it out of this mystery, somebody somewhere is doing this with my data, and it brings it to, this researcher at this hospital is interested in doing this because, and it will help me because. I think that goes a long way of just demystifying it and, having a frank conversation about the outcomes.
Fantastic. Thank you again. We're going to ask our speakers to have a seat, and we'll come back for the panel.
Great.
So thanks so much.
Thank you.
Really powerful. So I'll just make a comment about where we're going. So we had the big overview talk. We have this amazing story, and now we're going to zoom into some different conditions and thinking about how we think about that. And I think there's no one better for some of the genetics around aortic dissection risk than Dianna Milewicz, who you all know, I'm sure well, who's a cardiovascular chair at the Texas Health Science Center, has done a tremendous amount of research here. She's going to come up and talk to us about the predisposition to now, from Ehlers-Danlos to all aortic dissection risk. Thanks, Dianna.
Great, thank you. Hard act to follow, Katie. I'm going to talk to you about our research and into thoracic aortic aneurysms and dissections. And I love this sort of like graphic slide to indicate what we're talking about in terms of the genetic risk that Scott already nicely introduced. And at the one end of the spectrum, we have people where they get an alteration in a single gene in the human genome, and that confers a very highly penetrant risk for disease. And for thoracic aortic disease, right now our data say that that's about 20% of the patients. And some of them have syndromic features, but the majority do not. And then at the other end of the spectrum, you have people like DeBakey, who dissected, and Grant Wall, who dissected at the World Cup. No family history, no markers that they're at risk for dissection, and have gone on to dissect. And that's probably where this polygenic risk score comes in, but I think there's also a role for rare variants in that disease also, and probably a combination of both. But my research has really focused on going after these highly penetrant genes because they are highly predictive of disease, and thoracic aortic dissections can be completely prevented if we know who's at risk. So for a disease to study, this is perfect. If we identify somebody who's at risk, we have a way to treat it. So my lab and others have identified to date a large list of genes. We still have about 1,000 families in my study that are unsolved, and we only solve about 40% of the families. So we have a long way to go, probably another 60 genes to identify. But all these genes are now in diagnostic panels worldwide to identify people at risk for the disease. Now, so we have all these genes, and what does this mean to somebody who comes into the hospital with a type A or type B dissection? And if you go, and we've gone through and actually sequenced patients under the age of 60 that have no family history, have no syndromic features, and asked the question, what % of those patients will have a variant in one of these genes? And if you'll just, we did this twice in 2 different batches because I didn't believe our first results. And so we went back and did another set. But what we found out is that 10% of the patients walking in with a dissection will have a variant in one of these genes. The primary gene is FBN1, and that's the gene for Marfan syndrome. So there's a lot of people out there that have Marfan syndrome without syndromic features. And if you look, another surprise from this data is that the burden of rare variants is higher in people with descending dissections than ascending dissections. So if you take your, and that's a big surprise, if you take your Marfan patient, they have about a 10% risk for a type B dissection, but the other 90% present with a type A dissection. These variants are highly penetrant and highly predictive that if they don't have their aneurysm repair, they'll go on to have a dissection. So this is a bit of a surprise that there's such a burden of these rare variants with type B dissections, but also with type A dissections. So we've known about Marfan syndrome for 50 years, so we know how to manage that disease, but now we have this whole list of genes that we don't have much data in terms of how to manage the patients. And so myself and others launched the Montecino Aortic Consortium to collect data on these patients worldwide so that we could most efficiently and effectively begin to use precision medicine to manage the patients. So we launched this about 5 years ago, and I'm going to show you the data, some data from the first 1,000 patients in the study and just for select genes. So all these TGF-beta signaling genes that are grouped together are now called Loeys-Dietz syndrome genes. So we'll start out looking at 4 of those LDS genes. So here's a graph of the average age of onset of, a graph of age of onset of an aortic event defined as a dissection, type A or type B, or presentation with an aneurysm that needs surgical repair. And you can see already that between those different, these genes, there's a significantly different age of onset. First of all, if you look at TGF-beta2, by the age of 40, 50% have already had an event, but SMAD3, only 15% have had an event. So you're looking at 2 different risks, despite it has this one umbrella diagnosis. And also, if you look further, you can see in childhood, there are some variants in TGF-beta1 and 2 that cause childhood onset. But for the other 2 genes, there's no childhood onset. So this has lots of implications for imaging and how aggressive we need to be in childhood with these different genes. And once again, we're trying to prevent dissections, but also be able to efficiently manage these and cost-effectively manage these patients. And then if we just take one gene, TGF-beta2, and look at the individual variants that occur recurrently in this gene, you can see that we can even be more precise in terms of the age of onset. So now we have some genes that lead to childhood onset, whereas other genes lead to much later onset and actually aren't even completely penetrant well into their 80s. So, and so these have a lot of implications, and some of this was written into the treatment guidelines, the 2022 treatment guidelines, but we couldn't put all this data in. We tried to, and they actually cut it out because it would have ended up being half the guidelines. So we're in the process of trying to write gene-specific guidelines for this disease, but some did get into the ACC guidelines. These are surgical thresholds for these different Loeys-Dietz syndrome genes, and that varies based on the gene. And then these patients with Loeys-Dietz, there's a risk for going on to have aneurysms and dissections of the arteries, similar to what we heard with VEDS patients. So we've been comparing that risk with VEDS patients as part of the MAC consortium. And this is the data that we're coming up with. And some interesting things, like VEDS clearly has the highest, these are males, and men with VEDS have the highest penetrance and earliest events of other arterial aneurysms, and which is interesting that there's such a gender difference with VEDS, and we see that also with the aortic disease. So we can get, and now we can also begin to predict where those events will occur based on the individual gene too. And then we're also doing other studies. We're looking at venous disease. We're looking at other features of these conditions. But one thing we wanted to do is be able to use machine learning to look and see if we can define a facial profile that goes along with these individual genes. And so that if you see somebody with like Katie in the ER with a little neck pain, and thin skin, you can take a picture and have it say whether this is, has an increased likelihood of being VEDS or not. And so with the Loeys-Dietz syndrome genes, with Marfan syndrome, with VEDS, they do have a particular facial profile, and I can sometimes guess what the gene is, but could we do better with AI? And interestingly, we can. Here's VEDS here. Here's, I don't know, can't remember, these are Loeys-Dietz and Marfan syndrome, so there's some overlap, but you can see VEDS is very distinctive. And what's not shown on this is that normal children cluster outside of these patients. So this has a high potential to be able to quickly figure out if a patient that you're planning to put a TVAR in, or a patient that you were wondering whether or not they could have one of these syndromes, a quick way to look and see if they do. And then, so that's Loeys-Dietz and sort of an overview of what we're doing. And in the last few minutes, I want to talk about the genes that are involved in smooth muscle contraction, and in particular, ACTA2. With these genes, we can do the same thing, show different age of onset. PRKG1 is actually the earliest onset, and these patients dissect with neuroenlargement. So we're telling patients now that in their late teens, they need to start thinking about aortic surgery to go ahead and impair the ascending aorta, even if it's normal size, to prevent a type A dissection. So very aggressive management in these patients. And once again, we can take ACTA2 and show that there's some variants that predict childhood onset and need to be treated very aggressively. And with ACTA2, what we found was a very different story than you see with the Loeys-Dietz syndrome. There's a few variants in ACTA2 where if you have that variant, you're at a high risk for either early onset coronary artery disease or Moyamoya disease. And Moyamoya disease is a cerebral vascular condition where you have occlusion of the large arteries in the brain. And in these patients with ACTA2, Moyamoya can start as young as 16 months of age. So it's a very early onset. And it's just particular variants. So here we're getting down to the ultimate precision medicine in terms of there's only 2 variants that cause this Moyamoya-like phenotype. I'm talking about recurrent variants. There's some spotty variants that also do. And we have statistical evidence that 2 variants cause this early onset atherosclerotic disease. We've been working on this because to me as a geneticist and a researcher, this is fascinating. Why, when you mutate one amino acid in a smooth muscle contractile protein, do you get these other vascular diseases? And so we've been publishing a number of papers as we work on this. We're at the point of understanding the Moyamoya disease. We're at a point that we're launching a clinical trial. We have FDA and IRB approval, and we'll be starting our first patient within the next month. And we think this is a common pathway for all Moyamoya disease. So I'm actually meeting with the International Stroke Consortium to talk about launching the clinical trial using the same drug for Moyamoya disease beyond ACTA2. But I'm just going to talk very briefly about the early onset atherosclerosis. It's only in the last 5 years that we've really understood that smooth muscle cells play a major role in atherosclerosis, that they move out of the media into the plaque, and if you look in the plaque, 50 to 70% of the cells are smooth muscle derived. And it took a long time because the smooth muscles move into the plaque and they take on features of macrophages and other type cells. So it's not until we could lineage trace those smooth muscle cells that we actually determined this. And the ACTA2 is specifically expressed in smooth muscle cells. So the early onset atherosclerosis in this condition is driven by the smooth muscle cell. And here's a family with that arginine 179 mutation. And in red are the people with the mutation in early onset events. And you can see they're as young as 33 with no atherosclerotic risk factor. So we've been working on this, and I'll go through our data just briefly. It's kind of complicated, but in wild-type smooth muscle, if you have wild-type smooth muscle cells that are exposed to cholesterol, the cholesterol comes into the cell, drives ER stress, in particular PERK signaling, and that drives the smooth muscle to start migrating and also taking on features of a macrophage. And with the ACTA2, what we found was that the mutations that caused early onset atherosclerosis were not correctly folded, and that triggered cytosolic stress. And that led to activation of increased synthesis of all the enzymes involved in cholesterol biosynthesis, including HMG-CoA reductase. And that alone increased the amount of cholesterol inside the cell, drove smooth muscle modulation and migration, and increased burden of atherosclerosis. So this is a smooth muscle-driven disease, and we've been working on, is this a common, first of all, I think this, and we now know that if we target HMG-CoA reductase with statins, we're actually doing targeted therapy for this disease. So we want to start these patients at risk on statin, but we don't know what age to start them. We don't know if a calcium score would be a good way to figure out, because not all the patients get atherosclerosis, even with the variants that cause misfolding. And also this may explain why statins work so effectively, even in people that have a risk, but don't have elevated cholesterol. It may be through this pathway, and we're really thoroughly exploring that. We have data that we're getting ready to publish that homocystinuria drives increased plaque burden based on this pathway. And now we're working on hypertension, because it's already known that with hypertension, cell stress goes up in the smooth muscle cells. So look forward for more data on this. So things that I think that are raised by this, how do we effectively translate this data that we're collecting on these patients to busy physicians? And we're working on an app where you put in the gene and the variant, and it gives you the recommendations, but then you can look further and it gives you all the data that those recommendations are based on, because some patients want to see the data. And then we're also, I want to mention that I think clinical trials going forward really need genetic data, not only these rare variants, but that genetic risk score. I think the goal of my research, and I think probably Scott's, is we have to be able to identify people in the population that are at risk for dissection, and how do we get there? And we have no drugs that prevent dissections, and we have pretty lousy drugs that slow aneurysm growth, but even that data is very weak. And I have a Leduc Award focused on that, and now we have the MAC consortium to start those clinical trials. So once we have drugs, we're ready to go. And then I think there's a need for therapeutics that target, you saw that long list of therapeutics that Scott put up there. None of those target smooth muscle cells. And then we know they migrate, they modulate. If we block that, how much of atherosclerosis do we block? I can tell you in the mouse model, if we block the ER stress driven by cholesterol, we prevent 80% of the atherosclerosis. Okay. And I just want to mention that I have a large number of collaborators and funding sources. Thank you.
That's great. Amazing. Let me just see if there are any questions. I'm just going to raise that a sec. Any comments or questions from anything? A couple things. As you think about our morning, we're going from precision, I'll call it, to personalization in these two panels. We went from the overview, the patient story, now to the aorta, and a variety of conditions, and actually really exciting, interesting work of just beyond mechanisms we know as we think about these variants. Certainly the smooth muscle story is a very new story for us, a new target, new opportunities. And then we also saw just how one change of one amino acid can lead to such a different disease condition, which I think is also fascinating. But as we sit and think of all that information, there's going to be a lot of places for us to go. Any questions quickly? Diana's going to be back up for our, Katie, do you have a question in the back? Great. No, we have a panel. We're good. We'll take the questions when we get them.
Okay. I loved the slide on AI recognition of the three disorders I'm most familiar with. I was interested in, we know that participation of people of color is not high right now, and I think the recognition of people with these conditions who are Black or Hispanic or have non-white backgrounds is really low in research. I'm interested in how like what percentage of the patients went into this AI modeling that were not white, and how do you envision that moving forward to make sure that, because we don't know if the features are slightly different?
I agree with you completely, and I can't tell you off the top of the head what proportion were other ethnic and racial groups other than European Americans, but we are really pushing to include those patients and really trying to make sure we have a very diverse group of researchers that are involved in the patient interactions so that it's easier to get those patients involved.
One question comes from Matt, but before we get to that, just more people like Katie helping us to get more patient, people with these conditions to help from different backgrounds. What's going to help?
And just one note, we know Hispanics do not have the skeletal features that, in Marfan patients that are Hispanics do not have the skeletal features that we see in European Americans. So it's really important to know these things clinically. Yeah.
What a wonderful agenda to have you follow, our patient advocate. I'm a vascular surgeon, and in the last year, I referred a patient for genetic testing. It was a man with some unusual aneurysms. I was kind of plus minus about doing it, but he had other aneurysms in his family. I fixed his aortic aneurysm. It took him 11 months to get in and see a geneticist, and his test came back that he had vascular Ehlers-Danlos. And I was just going to ask, do we need to rethink the order of operations here? And I imagine, I don't know what it's like to practice as a geneticist. I know that at my institution, there's 100-fold more people that need to see a geneticist than we have geneticists to see them. Could we reorder this where the genetic testing occurs on the front end and the geneticist is only seeing needles instead of a haystack with one needle in it that they can help?
Yeah, that's a great question, and this is why we pushed the diagnostic tests that have taken the genes that we've identified into clinical testing to do direct-to-consumer testing. But it has to be bundled with genetic counseling. So anybody that thinks they have a mutation in one of these genes, or any patient that has to wait a year, can go to Invitae or GeneDx and just order it that way, and they can even pull in the insurance to cover it. But right now, the cost of testing for all these genes is less than an echo in terms of the cost.
Yep, less than an echo. We'll get back to in our panel, because there's a lot to be tackled with what you just said, which is the clinical pathway management information of genetic testing is a routine pathway, which I think is very important. Cognizant, thank you so much, Dianna, thanks. So we'll go to the panel because we'll have more. Sorry, Mark, I know you got a question, I apologize. So I want us now, we're going to zoom even further, or maybe similar in different directions, but Santhi Ganesh is going to be coming up. She's a professor of cardiology and internal medicine and human genetics at the University of Michigan, and she's going to tell us about gene discovery approaches, maybe even thinking about how we can use AI, biobanks, and consortia to do this, and use an example maybe that goes from rare disorder to aorta to hypertension. So we're getting into more population-based things. Santhi.
Thank you. Delighted to be here. Thanks for the opportunity to talk and be part of this conversation. So we're merging the prior panel into this one, but it's a bit of a shift where, the topic I was given is quite broad. Phenotypic expressions, relation to gene discovery approaches, what the potential is of AI, biobanks linked to electronic health records, consortia, and so forth. So what I thought I'd do is highlight some principles that might set up a good discussion on how we can use these approaches. So as part of my disclosures, they're listed there. I do have an invention disclosure on a genetic risk tool for fibromuscular dysplasia, which is one of the things I'm going to talk about. So I thought I'd describe some gene discovery principles. I have 2 slides that are a bit more didactic, and I'll keep it to that. Some phenotype considerations with the example of hypertension, and some current topics for gene discovery. So just very broadly, our goals in an association study are to identify associated genetic factors, loci, genes, define what the causal effects are, use this to understand functional relevance, therapeutic potential, but more recently also define clinical risks, and then use this for personalized medicine goals, as we've been talking about this morning. So This is the first slide that's just a general principle that's been touched on, but just so we're all on the same page, this is allele frequency, so how common is a genetic variant in the population, from rare on the left all the way to common on the right, and effect size for a given allele with kind of the scale of phenotypic severity of what we tend to see on the top and what our statistical power tends to be. So when we're thinking about setting up these studies, how do we want to put together a study design? So all the way at the left upper corner there is what Dr. Milewicz was speaking about with some of these variants that are high impact with typically a more strong phenotype, although there's a great degree of variability in how these present, as she nicely outlined. And here we might be thinking about pedigree studies looking at very rare variants, all the way down to the other end of the spectrum, common diseases, what Dr. Damrower was speaking about, common diseases, genome-wide association studies. These tend to be regulatory variants affecting how genes are expressed and when they're turned on or turned off. And the odds ratios tend to be a lot more subtle. 1.2, 1.1 might be what we're hoping to observe in a genome-wide association study. And then in the middle, more severe disease, the severe end of the spectrum for some common diseases, or for Mendelian disease, things that are a bit more common in the population and maybe have a little bit of a more subtle contribution to disease. And here we might be thinking about using exome sequencing or targeted sequencing approaches. So the second principle I wanted to outline, if we're talking about association studies, is really what are the power considerations. So the scale of what we're talking about, and I'll show some examples, one for a rare disease where we had 4 patients that made up the discovery pool, all the way to what you're doing in a GWAS, where ideally you would have hundreds of thousands of people, and now we have genome-wide association studies that have been done in 1 million plus individuals for certain traits. And what you're trying to do by increasing your sample size is effectively increase your statistical power. The phenotype considerations I think are super important here, where we're often dealing with heterogeneous traits. We might have what's called misspecification. I'll show an example of this. And there's a general principle that continuous traits have more statistical power than dichotomous traits. And I'll show an example of this. So also, just so that there's some kind of common understanding of what a genome-wide association workflow might be, if you have cases and controls for a dichotomous trait, you're talking about getting your samples, doing your genotyping either together or side by side ideally. Sometimes we borrow data sets from other places, put them together, do your QC, do what's called imputation to get in the variants you didn't necessarily assay for, but you can bring into your data set based on reference genome sequences. Generate your principal components, estimate ancestry, do all your matching, and then you do your association studies that are the output of, the associated SNPs and what makes up these polygenic risk scores. And I guess my only point here for GWAS is that these are very well-established workflows. It's fairly technical in certain aspects of what you're sequencing or genotyping and how you want to go about that, but they're all very solvable issues, and I think really where we should be focusing our time and what really makes or breaks a study is the phenotype considerations. And so what phenotype are we studying? Is it susceptibility, disease resilience perhaps, onset progression, and so forth, all the way to pharmacogenomics? Are we thinking about qualitative or quantitative measures under reliability and accuracy? Again, some of the things that I talked about, misspecification, how are you actually defining a case? How are you defining a control? How do you know a control doesn't actually have the phenotype? Ascertainment biases and measurement errors. So in cardiovascular traits, we're often adjusting for age and sex. We can adjust for ancestry to some degree. Think about intermediate phenotypes, and I'll touch on what that might look like. And there's opportunities now with large-scale genotyping, sequencing efforts linked to electronic health records to now get into sample sizes of millions of individuals. So that's exciting and also offers some challenges. So again, for hypertension as the example, when we think about hypertension in a population, it's really, you're on this distribution of blood pressure and you're at the high end of that spectrum. I would say diagnosed hypertension, if we look at the Venn diagram here, is probably like this, that there's undiagnosed cases, there's diagnosed hypertension that we know about. We're thinking about secondary hypertension, a lot of the work that we do in my own research program, and the specific kind of demonstration of that might be renovascular hypertension due to, in this example, multifocal stenosis. And then what Dr. Coleman will talk about is even a rarer subset with arterial fibrodysplasia and renovascular hypertension. Very low frequency cases, but very severe extreme of a phenotype. And so I think there are some examples to pull out from all of this that might help us with the discussion today. We might think about population samples for hypertension in the population. For diagnosed hypertension, we could go into the electronic health record and look for diagnosis codes. And then for lower frequency diseases, we're often looking at assembling our own clinical cohorts. And how do we go about that is a question. So the first case I'll show is diagnosed hypertension. So the first generation of genome-wide association studies, which again is you have 1 million markers across the genome, you do logistic regression at each of these sites, you do multiple testing correction for all the tests you've performed, and in this one it was about 14,000 cases for 7 different diseases and 3,000 shared controls. And really what you see are these green peaks up above, and like for coronary artery disease, the 9p21 locus that Dr. Dianna mentioned. Hypertension, however, was kind of a flat signal, and the discussion was why was hypertension amiss, and it's a dichotomous trait. There was probably a lot of hypertension in the population that wasn't diagnosed, so the controls, if your controls actually have your phenotype of interest, that's going to really diminish your power, and that's misspecification. So in a consortium objective, with the more recent consortia being things like all of the NHLBI funded cohorts have been genotyped, and then those data can be analyzed against the clinical variables and put together and meta-analyzed. And there are different ways to do this, but phenotype-focused cohorts, population cohorts are the kind of the NHLBI cohorts I was mentioning, and then biobanks linked to EHR with kind of this continuum of increasing sample size. And the objectives are to aggregate resources. Typically, we're talking about sample size, but there might be other resources as far as funding and so forth to try to put together these larger scale, well-powered experiments. Specialized patient populations, try to diversify our samples. And what I think is one of the really fun aspects of consortium work is the interdisciplinary interactions, where you have not only more samples, better diversity, but you have different types of people coming into the consortium with different types of expertise and different types of perspectives that can think about not only analytic approaches, but clinical significance and all the way over to mechanism. So the question of why study intermediate phenotypes for something like hypertension, that would be blood pressure is the definition of it. For an outcome like MI, myocardial infarction, cholesterol, blood pressure, a major motivation for why we're studying these is that we're worried about these kinds of outcomes. And so we tend to have better power in general. We have lots of examples of this, and in AI, the idea is that maybe we can have not just the regular clinically measured traits, but maybe there's some novel traits we could be measuring and actually getting continuous measures out of and using those for analysis. So for hypertension in the population, I thought I would just highlight one example where instead of looking at hypertension, we looked at blood pressure as a continuous trait. We had this nice assembly of the blood pressure data in the Framingham study, the ERIC cohort, many of those NHLBI cohorts with longitudinal follow-up. And we took a really simple idea, which was to simply average blood pressures over time. Blood pressure is a very noisy trait. It's a continuous trait, but it's super noisy, and that doesn't help. And we just averaged up 2, 3, or 4 measurements more than 1 year apart. And really what we found, I think is best summarized here, the results from single visit and long-term average blood pressure are highly correlated. But if you look at how many hits are actually popping up for single visit versus long-term average, it was many more hits that we found. And essentially it was a 20% increase in statistical power by thinking about the phenotype that way. And so there's all sorts of examples we could kind of align with this discussion. So in the Nurses' Health Study, they asked participants to simply say, what decile of systolic blood pressure are you in? And actually, you would think that's super imprecise, but the results aligned very well with the known blood pressure signals. And so, how are you getting your phenotype? How are you putting that together is just a concept I wanted to highlight for what we're talking about today. Okay, so For secondary hypertension, I'll just show an example for fibromuscular dysplasia, what we did. We had worked for about 10 years enrolling patients with adult multifocal type FMD, and we were very proud to have our first study combining with Cleveland Clinic samples to have 550 samples, the Michigan Genomics Initiative, a bunch of controls. We did our GWAS and we didn't find anything. So we said, well, this is a super small study. We didn't have a kind of lucky outcome where something with an odds ratio of 2, like what we've seen for spontaneous coronary artery dissection with a comparable sample size. We actually did find multiple hits. And so what you do in that setting is you go to other groups that are similarly trying to understand a disease and may have patient samples, may have done genotyping or fund the genotyping, put that together. And then what we had were our first 5 or so loci for fibromuscular dysplasia. And so, there's a lot of effort that goes into this sort of thing from, each of the groups deciding how they're going to come together, how they're going to collaborate, all the way to how people will be recognized and so forth. But it's very much worth it, and at the end of the day, we had enough sample size to find these things. So just to put our findings on this map here, for blood pressure hypertension, we definitely have findings for this very specific form of secondary hypertension, fibromuscular dysplasia, and renovascular hypertension. We have new loci, genes that were strongly prioritized statistically out of the data. And we actually, by studying, I mentioned 4 patients, had the first actionable finding, the clinically actionable finding for FMD in a COL5A1 gene. It's a specific 1 variant change, 1 nucleotide change that changes the amino acid from a serine to glycine in this gene and causes a very severe phenotype with dissections. And borrowing off of that, we looked at other less severe variants in COL5A1 and found if you have FMD and you have one of these less severe variants, you add that on top of it, and now you have about a 3-fold risk of arterial dissections, which is one of our worries in FMD. So that's an example of trying to risk stratify. The final example I'll show is outside of hypertension, but thinking about biobank data and AI in a published study. So AI, we've touched on this already this morning, but we're in this example, thinking about how we can ascertain a new trait. And so here, what was done is in a UK, it's called the UK Biobank Resource, 43,000 participants with a chest MRI. It was a single slice MRI. And learning was used to find these 2 circles for the ascending and descending aorta, define them, measure a diameter, and now what was used for analysis with a continuous trait of aortic dimension rather than simply aneurysm or not aneurysmal. And this is their Manhattan plot. They had many, many findings using this, and different findings for the ascending aorta and the descending aorta, some of which overlap, the majority of which didn't. These findings were aggregated for the thoracic aneurysm, thoracic aortic dimension, and looked to the prediction of aneurysm dissection in the high polygenic risk score group. The top 10% did have a very different curve from the remainder of the group. So lots of interest in applying methods like this. I think we've talked about AI to maybe even find what are the questions that we might want to ask. Here it's been very directed, very intuitive, and I think is a great example of where the group was able to leverage this into new understanding. So I'll stop my comments there, and hopefully that's helpful to set up some of the discussion.
Thanks, that was fantastic. And a great transition again, because we went from, sort of scale down to scale up and just looked at hypertension broadly, but with FMD and some interesting conditions. Any questions for Santhi? I mean, I think today we're learning a lot of different things, a lot of terms, a lot about genetics, but it just tells you the depth of what we're about to get information-wise, potentially even more on our patients. We've spent time on genetics, but we haven't yet even talked about patient preferences, phenotype, other data we'll have on individuals that'll get into it. And our next talk will sort of go into some of those because it's going to be around how do we use things that are in the medical record, how do we use other data that might be. So maybe we'll just go to that. Thanks, Santhi. So I'm going to have Kate McGinigle come up. She's great, and she's the Associate Professor of Surgery at University of North Carolina. And thinking about what we've been talking about, which is genomes and how do we find, we're talking about AI, we're thinking about different places we can go. She's going to give us a talk about how you might use some of those strategies for a different condition, but an equally important condition, CLTI. Thanks.
Great, thank you so much. We are going to zoom way, way out. and talk from a more public health standpoint, which is where my background is, and how I've chosen to use machine learning to approach some of the public health problems that I think exist in CLTI specifically. I've got a couple of disclosures, but really I just want to acknowledge UNC, my institution, their Center for Healthcare Innovation, for supporting some of the work I'm going to share with you, as well as the SVS. Let's see here. And so, you know, from a public health perspective, I've been really, you know, fascinated in watching all of this kind of genetic and molecular data evolve and all of these really complex methods evolve. And I was thinking to myself, like, we have to be able to harness this, because when you think about it, the data that all of us have floating around in the cloud is just massive. And so in our lifetimes, we're going to have terabytes of data per individual. And you know, best estimates are that about 30% of the determinants of health are based on the genetic stuff that we've been talking about. But there's a lot of clinical data that's available to us now with imaging and labs and just notes in the EHR that are also influential and can determine our health. And so I focused in there first, which turns out is terabyte of data per person, which is a lot. And it's really just scratching the surface. And I've started to work my way into the behavioral and environmental data that's collected on all of us. And I would love to talk about that more later, because this is really, I think, where the patient voice comes in and where some of the feasibility of treatment algorithms comes in. And it's really important to study that as well. And it really can affect what happens to patients more than any of the other things that I'm going to discuss. And so this is a pretty popular diagram. I just pulled it off the internet, but I think it's really useful to illustrate kind of what is AI. And, you know, as was pointed out earlier, people don't really trust it. There's a lot of confusion about the terminology. And so AI, at its most basic form, is really just how Amazon predicts what marketing ads it's going to give you based off your purchasing history, right? The machine is programmed to learn what you're buying and then predict what you might want to buy next. Getting down a little bit deeper and more complex is the machine learning. And so this is what I'm going to talk about. It's the methods that we use to make classifications and discover patterns in massive amounts of data that's way too heterogeneous to kind of look at a priori and develop hypotheses. And then later this afternoon, there's going to be some talks on computer vision and deep learning, and that's a totally different subset, not related to what I'm doing now, but it's very interesting and I think can be layered on and paired in with some of this work. And, you know, we've talked about this already today. In vascular surgery, especially in PAD, we are this very one-size-fits-all approach to medicine. And, you know, we've tried to do some cardiac risk stratification. We certainly have Wi-Fi stages that helps us understand what's going on in the limb. But these are just some very mild clinical features, and we don't really have disease subtyping. We don't have any biomarkers. And we certainly haven't gotten it to the point where we can stratify patients out to then really personalize their care. And so this is the goal. This is the goal. And the way that I've approached it is starting out with a cohort of my patients. And so I'm very lucky to work at a great limb preservation center at UNC. And you can see, though, amongst some of my patients, there's a ton of variation in their care. And a lot of this is warranted based on the clinical presentation, but a lot of it is completely unwarranted. And if you look at, you know, some of these in the middle that have about 1 million yellow dots, you know, these are people who got about 85,000 debridements, which is potentially unnecessary and we could improve upon. And so what we have are tools that are focused on limb features, right? Every single one of those patients has their Wi-Fi stage in the chart, and we take it every single time and we measure it like a vital sign. But what I need is evidence that incorporates that with everything else that's going on with them, the really complexity and the heterogeneity of their genetic makeup, of their other comorbidities. We have risk prediction tools, but we don't have anything like they have in cancer that tells us what to do with those patients with different risks. And, you know, we have things for the now, and then we're very reactive. We wait for something to happen, and then based on what happens, we make a new decision. But I want sequential, evidence-supported decision-making over time. And so these are kind of the principles, in my mind, of precision medicine. And using machine learning to get to this place, I think, has a lot of promise. And so, you know, for example, I affectionately call these my Skittle people. This is kind of what vascular patients look like when they come to our clinic. They're all a little bit different, but there's some kind of general groupings that we as clinicians know about. So we know people with PAD and diabetes are different than people with PAD and not diabetes. We know that if you look like somebody who may have been enrolled in the best CLI trial, which is truly our only pivotal level 1A evidence in this patient population, you should get a single segment saphenous vein bypass. But then that leaves all the people with the purple backgrounds that we're not really sure what to do with for giving the alternative therapies. And then we know from best CLI there was a really high reintervention rate and a really high limb loss rate. And so we don't really know what to do next, and we don't know how to vary what we do next based on, whatever other color circle happens to be around your patient that day. And so there's a lot of structure and framework that needs to be built up initially. And so first we need to stage patients better and decide what those colors actually mean in real clinical terms. And so that's some of the initial work that I've done, and I'll share you some of the results. And then we need to decide what are the actual key decision points and what are reasonable treatments at those decision points. Is it more watchful waiting? Is it an intervention? And these are things that seem impossible to think about prospectively, because if we use the kind of a priori hypothesis clinical trial paradigm that we're used to, it's going to be 300 years before we've run enough trials that may or may not actually be useful in sequence. And so machine learning takes the endpoints of existing data. So for example, patients with amputation-free survival versus patients who have survived with an amputation. And it works backwards. And it figures out why are those patients having the clinical trajectory they're having, and why are some not? And who's having certain complications and who is not? And this exists already in all of our EHR data. And so eventually, using this kind of backwards approach, you can fill in what you think individuals with different color combinations will get the most benefit from the first therapy, what their secondary therapies are going to be, to get whatever the optimal outcome is that you are optimizing for. And so, this is how, with fully realized precision medicine, we can learn from the data to kind of create treatment algorithms. And this is usually where people start to say, wait, this is a lot of black box, and we don't really understand what the computer is doing here. And so this is the black box of machine learning, and it's really math. And if you speak the language, then it's not nearly as scary. And I am going to give you an example here of supervised latent Dirichlet allocation, which is a type of natural language processing. But I'm not going to talk to you about the math. I'm going to talk to you about movies. And so what this allows us to do is what's called supervised topic modeling. And so say you've got a book full of all of the movie reviews, and you tell your machine to start categorizing these reviews, right? And so it will go through and systematically thumb through all of the different language that's used in the movie reviews, and it'll start to make categorizations based on words that seem to clump together frequently. And so through this, you might sort out that, these movies are drama, and these ones are sci-fi, and this one's fantasy. Okay, that's pretty great, except that's not supervised. That's just a machine starting to group stuff together. And that doesn't help you if what you actually wanted to know was who had good movie reviews and who had bad movie reviews. And so you put in a supervising term to figure out, okay, these ones were all, bombed at the box office, and these ones were good. But then this is where you have to be a human, and you have to be a good scientist, because when you go back and do what my favorite statisticians call a reality check, you realize The Godfather is actually one of the best movies that's ever been made. And so this has to be wrong. And so in clinical terms, this is like saying black women are 4 times more likely to have a leg amputation. And so the computer identifies black women as people who should just get leg amputations straight out of the gate with no revascularization attempt, right? And so that's a real fear. That's a real thing that can happen. And if you do bad science, that can happen. But there's tons of frameworks, and there's things in place that prevents us from doing that, even with machine learning. And then you can kind of show what's happening on an individual basis. There was a really nice slide demonstrating that earlier. And so, for me in PAD, Obviously, as much as I love movies, I care even more about people with CLTI. And so I used the EHR database initially, and I was able to validate a lot of this methodology in the PREVENT-3 data. I was really pleased to be able to do that project with Dr. Conti. And so instead of a movie review, I had the computer look through all of the individual patients' notes. And I specifically wanted to know about features that were related to PAD, like tibial artery, diabetes, gangrene. I think that wound-free survival is a really important primary outcome, not just amputation-free survival. And so, for example, I could care about that in this particular model. And then the computer starts to put together things that seems to clump people into different likelihoods of having wound or amputation-free survival. And it turns out in the UNC cohort and again in the PREVENT-3 cohort, the results were about the same, which also happen to parallel what we know as clinicians, which is really reassuring. And so in stage 1, these are patients who are likely to have had a lot of previous revascularization, who were smokers but did not have diabetes. And in 1 year, they had about an 8% chance of death and an 8% chance of limb loss. Stage 2 had a similar limb loss rate, but twice the chance of death at a year. And that was primarily driven by diabetes. You can see there's a bigger blue spike there. And then stage 3 are the people who have the more uncontrolled comorbidities and end-stage renal disease. And they are at a 20% chance of limb loss and a 20% chance of death, with just a 60% amputation-free survival rate at 1 year. And so this makes sense. And so I think this is a good way to codify kind of what we know and how we kind of practice. And it can start to standardize a lot of this care and starts to define what some of those colors are on the Skittle people. So we know how to classify patients better at baseline. And so, you know, oh, here we go. Sorry, I gave you all of that information already. And so really, you know, what's next for me using a lot of this methodology is now that we're starting to define not just the patient's limb-based features, but their whole body features, I want to describe what happens to their trajectories over time and why some people are able to get one intervention and maintain their medical therapy and do really well, and why some people get a lot of interventions but still have an ideal outcome, and some people get a lot of interventions that were futile. We need to understand those trajectories. We need to understand the combinations of medical and surgical treatments, which again, it's a lot easier done retrospectively with the machine. And then one thing that I'm really, really excited about and passionate about trying to get started is involving more patient-reported outcomes and qualitative studies to understand the patient's perspective on barriers and facilitators to care. Because if you create an optimal treatment algorithm that nobody can adhere to, you're not actually moving the needle. And we've got some really great methods developed to attack this problem. And so then, in this kind of socially conscious way, we can develop, you know, clinically evidence-based treatment algorithms that are achievable, that can have genetic information layered in when we get there. And you get these treatment regimes, right? Which then, of course, because we do good science, we study prospectively through something called a SMART, which is a sequential multiple assessment randomized trial, which is it's a multiple randomization trial, so you can study treatment sequences instead of just one treatment at a time. And that's really useful for folks with chronic disease who are really heterogeneous. And this is how you confirm that your computer was giving you the right information as you were creating these algorithms. So that's my next number of years, and you can see how you can apply this to pretty much any disease state. And thank you very much.
Thank you. I know we have one more talk to go, but great conversation. Took us, I like that photo too at the end. Takes us from, you know, lots of sources of data. We've talked about genetics, but just the sheer amount of information that you're talking about that we're going to be generating, not just in the EHR, but others, is going to be really powerful for us. And then I think another stream of conversation that we're going to have. So far this morning, powerful patient story led to us to thinking about genetic syndromes from very monogenic, or I'll say Mendelian, to very polygenic risk scores. And we saw a condition like hypertension. I'm now going to get our I think, probably our last speaker for the morning before we go to coffee break and come back for the discussion. It's really exciting to introduce Dawn Coleman, who we were lucky enough to get to Duke to come and be our chief of vascular surgery and endovascular surgery. And she's going to talk to us about the molecular and genetic evaluation of pediatric kids. You heard a little bit about this in Santhi's, a specific population with renovascular hypertension.
Thanks, Dr. Patel. Thank you, Dr. Conti. Thanks, really, everybody who's made today possible. It's such a stimulating set of discussions so far, and I'm really so delighted to be a part of today. We're going to move gears, and hopefully it kind of ends up being full circle, thinking about the different levels of consideration. And I'm going to focus in today on a really rare, almost ultra-rare disease phenotype. And I apologize, my disclosure slide didn't make this slide set, but I have none, no financial disclosures at least, but I am funded by PCORI with our collaborative team. I'll share a little bit about that with you today. And I also, while I've recently relocated to Duke and I serve as the division chief there, I've retained an adjunct appointment at the University of Michigan. And I do think that's important because we're all really deeply committed to collaborative science, which I propose to you this team scientific approach is so requisite, especially in the space of rare disease, to address some of the gaps in knowledge that we have. I'm going to keep a bit of a clinical focus because the audience I know is heterogeneous as the patient phenotypes are that I'm going to share with you today, and I just want to give everybody a little background. But we're going to talk about pediatric renovascular hypertension. And what I'd like to share in these first few slides is really what a heterogeneous diagnosis this can be, which makes phenotyping sometimes a little tricky. So I'll give you a range of angiograms and patient stories, and we'll move forward from there. And this is one that I open up most of my talks with because it was one of our more striking phenotypes that did really well with surgery, and that's not always the case when these children are really small. And this was an 8-month-old that transferred from the Pacific Northwest to the University of Michigan, directly to the PICU. She was 8 months when we received her, and she had been completely healthy, no gestational complications, born term, discharged from the hospital day 1 with mom, but did have failure to thrive. And at some point, mom identified around 7.5, 8 months of age that her legs were blue and she was really fussy. And this really happened out of nowhere. And the child was brought to her pediatrician and noted to have both a cardiac murmur and severe hypertension that was really only present in the arms and not in the legs. Systolic pressures for this 8-month-old were over 200, and her diastolics well over 100. And a very appropriate and robust workup ensued, ultimately revealing a few findings that I'll highlight for you. Importantly, the aorta is occluded, really at the level of what you can see as the left renal. Her SMA has an ostial stenosis. Her left renal has an ostial stenosis, and you don't see the right kidney. And in fact, the right kidney shown by the arrows atrophic. It's really not vascularized at all, and the circle highlights that left renal ostial stenosis. So there's nothing I like worse than operating on babies. It's my least favorite thing to do, and it's very scary. And none of us like operating on babies, turns out, not even my friends at Boston Children's. We do everything we can avoid to operate on babies. And we really do aggressively medically manage these very young children with this disease because of the implications intrinsic to their small size and challenging anatomic features. And we loaded this little girl up over about 4 weeks with an aggressive medical management approach. We had her on 5 or 6 drugs, alternating with drips. And we found she had this really narrow sweet spot, and if we dropped her pressure too low, her kidneys started to fail and she had belly pain, probably from mesenteric ischemia. And ultimately, she went on to have progressive cardiopulmonary failure. She was intubated, maintained on a ventilator, and at 9 months, we took her to the operating room to revascularize her. We did that by way of an aorto to aortic bypass, which you can see here coming off the supraceliac aorta. This is a prosthetic bypass. We took her right kidney out. We reimplanted the left renal artery onto this stump of aorta, so it fills now in a retrograde fashion. And she did super. She left on day 18 with a single drug. Her blood pressure was adequately controlled. Her kidney function was normal. And at 1 year, you would never have guessed that she had had this effect, short of being on one medication for blood pressure. Everything remodeled favorably, and I think that's important. Her single kidney hypertrophied, her heart positively remodeled, and the LVH that she had had on presentation recovered, and all of her failure to thrive went away. She was in the 86th percentile. And I show this picture really just to highlight two things. One, her small size, but importantly, the incision we use. I like this, and I'm going to affectionately refer to it as the upside-down Stanley smile from the University of Michigan. And then this is her last fall at the big house. She's my son's age, and she's done beautifully. But I will tell you that what lingers in my mind when I think about this little girl is I suggested to Mom that it would be likely she would require a reintervention down the road. We didn't know when that would be, we didn't know what that would be, but Mom's always waiting for the other shoe to drop. And so at some point, this little girl will need another operation. And not being able to really tangibly predict that for her makes me feel like I'm failing this patient and her family. And the parents are desperate to understand why this happened. There's a lot of maternal guilt. I've heard paternal guilt that I've heard from families. And as I think has been hinted at in the morning, when you have a rare disease and you have data and you don't always know what it means, it's a really uncomfortable place to be. So I'm 100% confident we can do better. Here's another phenotype, a more subtle aortic coarctation, essentially with no kidneys filling, but on delayed imaging, here's reconstitution of a nearly normal main renal artery on the left. This is a child from Israel that had a right nephrectomy and ended up on dialysis after a respiratory infection. And the renovascular hypertension element had been really unclear up until that point. This kid transferred to the University of Michigan to have an aortic repair so that he could get a transplant back home. We ended up reimplanting his renal, patching his aorta, and he came off dialysis and was down to 2 drugs for antihypertensive therapy. And here's another really remarkable phenotype of a long segment thoracoabdominal aortic occlusion. I like this picture. This is a 5-year-old. There's just no aorta, but these beautiful collaterals, and it really goes to show in part, which we'll talk about, these kids are supremely well compensated. So delays in diagnosis are super common because they often will lack symptoms. And this was a fun multidisciplinary case with a congenital cardiac team at the University of Michigan where her renals didn't need a thing and all we had to do was bypass her aorta off the ascending. And then finally, a neurofibromatosis case. And I share this intentionally because we're going to talk a lot about this phenotype. But here's a teenager, only 2-drug hypertension, which technically doesn't even meet my criteria to offer surgical revascularization, but she has these really complicated lobular aneurysms off which there are these 3 branches going to her gland. And so the indication for treatment in this particular case is the aneurysmal phenotype and the risk of thromboembolism and rupture. This patient was treated with an ex vivo reconstruction. We syndactylized all 3 of the outflow branches into 1 and then did a bypass off her aorta, which you can see here, and she did great. But here's another kid with NF1 who doesn't have any aneurysms at all, but a subtle coarctation and bilateral renal artery stenoses. So different variants, and often patients with no variants, can look the same. And the question really is, are they the same, and should they be treated or considered the same? When I trained with Jim Stanley at Michigan, I was made to assume that the Michigan way was the right way. If you did a perfect operation, your patient should be cured of hypertension, and you should cure 90+% of kids of their hypertension. And that's what I learned as a fellow. And maybe that wasn't quite the messaging he wanted to relay to me, but that's what I received. And I started feeling like a really terrible doctor because we were not curing 90% of our kids with hypertension. And so that matters. This is a rare disease. Renovascular hypertension, all comers, is about the 3rd most common cause of hypertension in kids. We think it accounts for maybe 5 to 10% of pediatric hypertension. After intrinsic renal disease, after thoracic coarctation, the workup for secondary hypertension is really, really broad. We do know that up to 40% of patients may have a genetic etiology. Some of these has been hinted at today, but I'm going to focus again on the NF1 phenotype and genotype because for us, it's the one that we have seen most frequently to substantiate some of our conclusions. And importantly, renovascular hypertension simply means that the kidneys feel a suppressed blood flow pressure and they activate the renin-aldosterone system, in which case there are multiple pathways by which blood pressure is elevated and extremely hard to treat. And it can be simply from a renal artery narrowing, which is most common in our adult patients, But we find in these kids that mid-abdominal aortic syndrome is a really, really important disease subtype, which is classified by the most cephalatic stent of the aortic narrowing, which is almost always above the renals. It can go all the way up into the descending thoracic. Renal involvement is almost always present at 90%, and the celiac and SMA are narrowed about 2/3 of the time. And I used to hear this described as a developmental misadventure, where during the 4th week of fetal development, embryonic fusion of dorsal aortas was flawed. And you see some other really interesting things around the aortic branches, multiple accessory renals on both sides, really deranged looking lumbars. And so there probably is something to that, but why? These are our criteria for revascularization, but these are ours. Like, this is how I take care of patients. And I'll say that it made a lot of sense a decade ago when I started this practice, but it has become more individualized since we've learned more about these patients clinically and scientifically. So we have a rare disease with a heterogeneous phenotype, and I thought I was doing a crappy job with surgery. So we really dug into our clinical data to understand if there were clinical features or predictors that we could associate with better or worse success metrics. And that raises a really important question about how we qualify a treatment success or failure. And it's probably more complicated than it was when I thought about this 10 years ago. And we identified a few things that I think are important. The first is that our patient population at Michigan was extremely complicated, and more so than it had been in decades past. So we found that about 20% of our kids had neurofibromatosis. 45% had abdominal aortic coarctation, and I would say that is much more severe than what you would see if you looked at all renovascular hypertension patients. If you look at other centers of excellence like the Children's Hospital of Philadelphia or the Hospital for Sick Kids in Toronto, they have a far more significant presence of kids with mid-distal segmental renal artery stenoses, WEBs, that classic FMD, or pediatric FMD as they're referred to, than these mid-aortic syndrome patients. And so I think our surgical-centric practice really biased the data and the patient population that we had. But we also found that we were seeing a lot of treatment failures. 1/3 of our kids had failed some prior intervention. Here's an example of what some of these renal artery lesions can look like. With, in our series, osteal disease being most common and almost consistently associated with the mid-aortic phenotype. And bilaterality was really common. We offered a handful of surgical procedures to these kids, and I want to spend just a second talking about this, not because I expect you all, especially the non-surgeons, to leave knowing how we should treat these patients, but to propose that we don't actually know how we should be treating these patients. At the University of Michigan and now at Duke, we're going to offer mainly renal artery reimplantations or renal artery bypasses to revascularize based on the decades of practice legacy that Jim Stanley accrued over time. It works really well. But other sites will have simply a urologist or a transplant surgeon, and they're doing auto transplants. And some people are being more and more aggressive with catheter-based approaches, and that doesn't make them wrong at all. In the aortic space, we're offering patch aortoplasty and aortic bypasses, as I had hinted at, with PTFE. And I'll tell you, that's not at all what they're doing at Boston Children's. They're taking meandering collateral mesenteric vessels and bypassing with autogenous conduit. They're putting tissue expanders behind the aortic to allow it to lengthen so they can do a resection and reconstruction, and that's super novel and innovative. And it's what they're doing, and it's not what we're doing. I don't think there's much of a role, at least right now, with current devices for endovascular therapy in the aortic space, but it has been described. My point being that patients struggle when they get multiple opinions and they hear, well, Dr. Coleman and Dr. Eliason were going to do it this way at Michigan, but this is how they're going to do it at Boston, and what am I supposed to do? And we don't actually have any good data that suggests one treatment approach is better than the other. When we looked at our data, and it goes back, about to kids treated since the early 90s, 1991. There were about 170 kids treated. We found a 20% risk of reoperation down the road. And this is a super important slide. Secondary interventions to preserve primary patency were required in 13% of kids, so 22 of those 35 children. In the other kids, A secondary intervention was required for disease in a completely different vascular territory. When the kid was 5, I fixed the right renal, and at 10, he needed the left renal fixed, but that left renal looked normal 5 years ago. Or we fixed a kidney vessel when the kid was 6, and then they're a teenager, and all of a sudden they have a coarctation, and now there's a pressure gradient across their aorta. So that's super fascinating. And our improvement benefits were really blunted in these kids compared to Dr. Stanley's practice. We identified with logistic regression modeling that younger age at operation and the presence of mid-aortic syndrome seem to predict reoperation. In fact, if a child was 1 year older, every 1 year increase in age resulted in a reduction of 10% in the need for reoperation. We also found that NF1 was negatively associated with hypertension benefits, and importantly, prior interventions were the single independent factor predictive that somebody undergoing a remedial operation was not going to be cured of their hypertension. This was our team at Michigan, made up of all of these specialists, and I really don't have everybody's pictures. It's beyond the scope of what I can share today, but I do want to highlight my partner, former partner, John Eliason, and my teacher and mentor, Jim Stanley. And I want to share with you a little bit around how the scientific and, importantly, genomic and molecular considerations have shaped how we care for these patients. This was our mission at Michigan. And importantly, we highlighted within this mission statement cutting-edge laboratory and clinical research. And that was 5, 6 years ago. And I was perfectly partnered with Dr. Santhi Ganesh right out of fellowship. And she was a mentor to me, scientifically speaking. She's become a really critical collaborator, partner, and friend to kind of help work the space. And I think you can understand from some of what she shared how our paths crossed. We looked at these lesions under the microscope, and I love this picture. I show it to you for a couple reasons. But one, all of this blue is a trichrome stain that really showcases the collagen content with these intimal plugs. There's fragmentation of the elastic lamina, there's medial thinning, there's excessive periadventitial elastin, and these vessels are small. So they're smaller by age than they should be where they're narrowed, meaning this is not just an intraluminal process. And if this line represents over age in years to 16, the angiographic diameter of the lumen of a normal pediatric renal artery, here's the luminal diameter in these kids under a microscope, and this is the outer diameter. So they're small vessels, never mind all of the other cellular derangements that we see. I already referenced that there's a genetic etiology. Up to 40% of these cases, it's been published through the Boston Children's Group and the Great Ormond Street Hospital Group across the pond. And we looked at ours, and when this study was published, we had sequenced 102 kids. And I share with you really today just data around our NF1 variants. And here they are. Oops, sorry. And importantly, we identified 10 novel NF1 variants. And I went into this work with Dr. Ganesh thinking maybe there's a genotype-phenotype. Maybe we can identify a series of variants that portend a risk of arteriopathy, and that becomes potentially a screening tool for these kids. Spoiler alert, we didn't find that yet, and maybe it's just because we're underpowered, but NF1 is such a huge gene, and major deletions have been associated with other phenotypes, specifically in kind of the tumor space, different syndromic facial variants, and some behavioral disturbances or developmental delays. Here's what we did find, though. We enrolled and continue to enroll patients in a clinical database. We get their blood, saliva, OR tissue, and it's available. And we've done whole exome sequencing, RNA-seq analysis of the NF1 MAPK pathway genes and NCP1 levels. And we've identified a few things. The first is that the NF1 gene expression from resected dysplastic arteries is significantly reduced in pathologic tissue by 5-fold. And globally, several genes have been observed to be differentially expressed in this tissue. This volcano plot specifically demonstrates differential expression, and notably CCL2, which is also known as MCP1, this is an inflammatory marker for tissue injury, is significantly upregulated in these patient tissues. And importantly, the gene set enrichment analysis revealed significant upregulation of MAPK-related pathways. And to validate this, we looked at phospho-ERK expression through immunohistochemistry, which you can see here. And essentially, we found the cytoplasmic and nuclear immunolabeling for phospho-ERK to be 4 to 20-fold higher in the media and intima of our NF1 patients. So we made a couple hypotheses, pseudo-conclusions, hypotheses from this data, but we believe that the increased level of inflammation and the upregulation of the MAPK pathway may suggest the downstream effect observed in these NF1 variants. And we link that then to understanding that NF1 is associated clinically with a more aggressive adverse vascular remodeling process, and we hypothesize that this contributes to the treatment limitations we've seen in our clinical data. So we can do better. And based on all of the things I've already hinted at, with low disease frequency, the expansion of techniques that are inconsistent across centers, patients and families are challenged to balance differing sets of recommendations, and we really don't know what the optimal treatment is. And importantly, access to care is challenged because there aren't a lot of teams taking care of these kids. We sought to do better. And Dr. Ganesh and I were funded by a Michigan Taubman Institute Precision Health Award. And in the pediatric space, that really came to fruition with an international symposium in 2019 that I share with you here. We were really intentional to reach out to people that had been maybe viewed previously as competitors, but really have become, importantly, collaborators in this space. And we brought together 40 guests from 15 centers, 6 countries, and 12 specialties to really think about this disease with some intentionality. And ultimately, this yielded overwhelming support to work proactively together, and we've since been funded by a PCORI Engagement Award for capacity building. And our membership now is at 205 strong, and we are actively collaborating with our patients and families as research partners to prioritize where our efforts go. I'm not going to get into some of our early work from the collaborative because it might be a little bit beyond the scope of our time right now, but I will say a few things. Listening to our patients and our families have allowed us to shift some of our research priorities. And I went in with a vision, and I'll tell you it looks very different now as influenced by the partners we get to work with, listening to what they need. And we've prioritized 12 clinical questions with the support of our stakeholders, and we're working on that now. Our long-term goals with this consortium work are here. We aim to accomplish deep phenotyping through multi-site registries, cohort development, image repositories to define disease subtypes, define natural history better using data science methods that integrate a lot of clinical data, but also patient-reported outcomes, as we've talked about, laboratory, imaging, environmental, and omics data. We're looking to elucidate these genotype-phenotype interactions, ideally to develop reliable and valid predictive tools that will help us determine who's going to respond to the best treatment and encourage innovative methods to optimize patient access to care. And finally, we'll have a group of patients and families standing by for ready activation for pragmatic interventional and clinical trials. And so we've got a lot of work still in front of us, and I think the patient engagement piece is a really important one maybe we can talk about during the panel. I was asked to share our 3 prioritized research questions in my slides, and I'll share them here just for you to look through, but it really has to do with how we can do better for these patients, getting them sooner, treating them sooner. How do we treat them, and how should that be influenced by phenotype, genotype, and how can we alter the natural history, both pre-surgery and post-surgery, so we can reduce the reoperations that are required? And so with that, I thank you.
Thanks, that was great. And any questions or comments before we go to break? I know we promised everyone a break. A really powerful story of how you can take, yeah.
I'm very fascinated, dealing with PAD, the problems and complications with neointimal hyperplasia. And what you showed there in these kids and the renal arteries, that was definitely, hyperplastic intima. Have you looked into correlations in what's going on there compared to other instances of neointimal hyperplasia biology or
Yeah, it's a really great question. I don't know how much we can get into that now, and it may be better discussion to engage Dr. Ganesh with, because It also is an interest for her, given her adult work in the past. I'd say at least a couple things, and I can at least speak to the clinical element. We see robust tissue remodeling that's very negative after intervention. So despite a flawless post-op angiogram, these kids will accrue really rapid neointimal hyperplasia at sites of vascular injury. And so there is something to this. And when I talk about altering the natural history post-operatively, my sense is there is more of a chronic inflammatory milieu than we've previously attributed to this diagnosis. And surgery, like in many vascular pathologies, is not really curative. We're putting Band-Aids on patients. So I think there's a lot to that, and it's probably more than we can talk about right in this moment.
Yeah, I think it's a great question. And when we get into our panel, we'll talk a little bit about how do we take what I'll call ends of phenotypic expression, kids with something, and say, how does that teach us about something that happens in adults undergoing some sort of a revascularization? Because the process, whether it's in the periphery or the coronary or any vascular tissue, the intimal hyperplasia has certainly been studied a lot. Dianna, one more question?
Just one quick note. Almost every gene that causes this phenotype also causes moya-moya disease.
Yeah.
And I actually called up Stanley about 10 years ago when we started finding moya-moya disease genes and said, can I sequence your cohort? And he goes, that's a good idea, but I think we'll do it here. And that's why I said, we'll talk to Santhi.
So we see moya-moya a lot.
But we do have a clear pathway for molecular pathogenesis of moya-moya. The lesions look the same. The neointimal hyperplasia is the same. It's all smooth muscle cell derived. The endothelial cells don't proliferate and fill up the lumen. It's the medial smooth muscle cells. And I'd love to talk about our clinical trial with you and Santhi and whether we should try it also in this disease.
That's a great point. And much of the biology and pathology, as you're talking about, for a long time around the smooth muscle response to intervention has been discussed. And so how the smooth muscles respond to any intervention is a critical one, and I suspect it's on that spectrum again. We promised you coffee. I've been told the one thing I was supposed to do is stay on time to get to coffee. So I want to be sure you all get out to the coffee break. Just to be clear, it's from 10:30 to 10:50. Come back here. Mohamed's here. We're excited. So we're going to have him give his talk, and then we're going to actually have our panel. So thanks, everybody, for a great morning. All right. So we're going to get started again. Thank you all for staying engaged. These, as we know, the coffee breaks are where some of the most fun stuff happens. So we'll try to make sure we have time to engage. We have one more talk, which we're going to do to round out the morning, and I just want to specifically, again, thanks to Mohamed Zayed, who did a lot through a lot of travel and other things to come here. But he's the Associate Professor of Surgery, Radiology, Molecular Cell Biology, Biomedical Engineering. I just got to meet him recently, star in vascular surgery. He's going to tell us a little bit about, we've talked about genetics, we've talked about AI, we've talked about personalization. Now we're going to think a little bit, at least, to think about some serum biomarkers for PAD, which would really help us. And we heard a call for these biomarkers and ways to get it. So, Mohamed, thank you.
Thank you. Thank you so much for this opportunity. And I'm sorry again that I missed dinner last night. I did have some issues getting in. It just wasn't meant to be. 2 flights having mechanical problems just wasn't meant to be. But vascular cures is near and dear to my heart. You know, I was a Wiley Scholar back in 2015, time flies, but the Vascular Cures Foundation and the Wiley Scholar was a huge catalyst to my professional career. And, you know, Vascular Cures is near and dear to my heart for many reasons, but particularly because of the early stage support that I received from the foundation. It made a huge difference. And actually, what I'm going to talk to you about today is actually something that stemmed from the work that I had done back when I was a Wiley Scholar, starting out my professional career and thinking about what am I interested in studying? How am I going to link this back to my patients? How am I going to link this back to my clinical practice? And I'm going to tell you a little bit about where we can potentially have an opportunity as vascular innovators that are linking with both academia and industry. And some of this actually I talked about at Duke when I was there just a couple days ago. So as vascular specialists, we all know that the predominant distribution of the disease is in 3 main vascular beds: the coronaries, the peripheral arterial system in general, that includes also the visceral system, just like Dr. Coleman mentioned, as well as the neurovasculature. And this leads to myocardial infarctions, chronic threatening ischemia, PAD, as well as stroke. So our mission here is to try to figure out a way to be able to make dents in these clinical manifestations of atheroprogression or occlusive disease in the vessels. This is a great article that was published a while ago, but it's still very relevant. It was an article that was multinational, and it looked at how are we doing in terms of treating patients with atheroprogression. It was a progress report, so to speak, over a 10-year period. And one thing that was really amazing about this article is that it stratified patients based on disease patterns, including risk factors like diabetes. Now, we all know diabetes is a major cardiovascular risk factor, but when we look at this data, it's really interesting. There's a couple things that are very important. One, you can see that over a 10-year period, we did fantastic in terms of reducing rates of acute myocardial infarction and stroke. These rates really decreased in patients with diabetes. It's really impressive, nearly by 50%. Now notice, amputation rates are relatively stable, right? They're stubbornly high still in these patients. And in fact, when you look at the y-axis here, between patients that don't have diabetes and the y-axis here of patients with diabetes, it's 10 times more, right? And that's over a 10-year period, a decade of research, a decade of clinical therapeutics, improvement in guidelines, the use of statins, and other therapeutics. When we look at our guidelines, they are fairly complicated, right? This is from the ACC AHA guidelines, but one thing that you'll notice is that these guidelines usually will start with an assessment of the serum lipids, right? That's usually the primary prevention strategy that we take as clinicians in terms of stratifying patients according to disease. Yes, they could be diabetic. Yes, they can have hypertension. Their age may differ, but our first and primary thing that we usually do is we draw some labs and we look at their lipid profile. And based on that lipid profile, we stratify them based on the severity of it, and then we decide what to do next. And you can see throughout these guidelines, actually up here, we'll look at the higher risk patients according to their LDL. Even the lower risk patients, we look at their LDL. And we stratify them further based on that. Well, let's take a look a little bit at this. We've assumed that this is a major way or major determinant of disease patterns. Why is that? There's a historical context to this. Michael Brown and Joseph Goldstein discovered the LDL receptor in 1985 and received the Nobel Prize for this. And if we are to look at the biochemistry of this, just to remind you, the liver synthesizes lipids. It makes lipoproteins. The lipoproteins are released into your serum. Your body utilizes it as an energy source, and VLDL particles are then broken down into LDL particles. LDL particles are then reabsorbed by your liver through the LDL receptor and then recycled, and so on and so forth. Now, if you have a defect in this LDL receptor, what does that give you? It gives you familial hypercholesteremia, right? And there's various variants of this, and that results in higher than normal lipids, higher than normal LDL, and higher than normal risk of atheroprogression. If you have a native elevation of LDL, you have a normal receptor, but you have higher LDL in your circulating serum, you're also at risk of atheroprogression. And this has been correlated with coronary studies as well as stroke studies. And so as such, Pfizer and others have taken advantage of this and have developed direct inhibitors to LDL functioning and reabsorption. And so the question is this really where we should be focusing on or not in terms of PAD? And I'm going to stand here and I'm going to try to challenge that notion, and I want us as vascular innovators to think about this a little intently. If we look back at the major studies that have evaluated lipoproteins and their risk of atheroprogression, surprisingly, none of these studies have found that lipoproteins, including LDL, triglycerides, HDL, or total cholesterol, have any correlation with incidence of PAD. Do you find that to be surprising? I do, right? Because you would assume that PAD and coronary disease and neurovascular disease are the same mechanism. But it looks like when you look at all these studies, including the Framingham study, the Framingham Offspring, the Annenberg study, the Physicians' Health Study, the Women's Health Study, the Health Professionals Follow-Up Study, and the Speedwell, none of these studies found any correlation with these lipoproteins and incidence of claudication, chronic limb-threatening ischemia, or the incidence of amputation. In fact, there's not a single prospective clinical randomized trial that has randomized patients to statins, for example, or no statins, that has found that this impacts amputation rates. Again, do you find that to be surprising? I do. Why is it? Well, the question is, and as I discussed with Dr. Patel recently, I think it's a different mechanism. I think that while LDL does confer risk for coronaries and for neurovascular disease, it probably does not confer risk factors for PAD. Okay, so that is the question, right? Now, how do we try to find a solution for this question? The question now is, can we find a more sensitive method for screening and diagnosing of PAD? If LDL is not sensitive enough, based on the prior studies that we just looked at, is there a better way? Because that would be essential for primary screening and prevention of PAD. Just fundamentally, if we're going to be going by the guidelines, we need to have a way to be able to identify individuals with PAD, particularly if they're asymptomatic, meaning that they have PAD, they don't know about it, but there's a potential opportunity to be able to decrease that risk of progression in that disease pattern. So our team at WashU and many others, including individuals in this room, have been trying to develop primary prevention tools for screening for PAD in various forms, right? Using molecular imaging, point-of-care perfusion, dynamic simulations, tissue profiling, as well as looking at the serum and plasma biomarkers. And so what I wanted to focus on for the purposes of today and to just incite discussion is the serum and plasma biomarkers. If we were to try to find a better serum and plasma biomarker that's more sensitive for PAD, how would we go about it? Well, one of the things as clinicians is our access to patients and our access to tissue. And if we're surgeons, also our access to plaque and diseased tissue. That is a huge, huge advantage from a clinical studying standpoint. As we see these patients, we need to be able to consider how can we potentially recruit them to mechanisms where we can collect very valuable serum and tissue substrates. In addition, we should be looking at existing clinical trials that are already recruiting patients, whether it be patients with PAD or other forms of atherogression, and see what resources are already being collected from these types of patients. And in addition, there have been already studies that have been very instrumental in making observations about atherogression that have already collected tissue and serum specimens. The question is, can we tap into these resources as well? And in doing so, can we be able to go back and look at the status of these patients and their PAD? Are they asymptomatic, symptomatic, or have chronic limb-threatening ischemia? What kind of specimens do we have access to, either in a retrospective or prospective fashion? And then finally, how can we link these back to the patient's data variables, for example, their calcium coronary scores, their lipoproteins, their ABIs, and other tests that can help validate their level of disease. And typically, the way that we think about this in academia is that we have a great idea, we develop that idea, we write a research grant, and we make discovery, testable hypotheses, and we publish it. And then we continue to do the same. But that is not our end game. Our end game is to try to translate these into practice, right? As vascular innovators, working with the vascular cures, we have an opportunity to be able to not just focus on this cycle, which is wonderful from an academic standpoint, but it will allow us to be able to bring these discoveries and these questions and these novel ideas into actual clinical practice. And it helps us as a team of specialists to be able to consider how do we collectively work with one another to be able to bring these ideas to fruition? And to what extent should we be patenting these ideas, licensing these ideas, and potentially even developing them for commercial purposes? How do we commercialize these ideas? And in order to do so, it really depends on building an entire ecosystem of innovation. And this is something that I think the Vascular Cures has an opportunity, along with many of the people in the room, to be able to build components of IP, publications, education, having a pipeline for developing proof of concepts, prototypes, testing capacity, and then being able to collaborate across the bridge with industry for licensing, seed funding, finding incubators or accelerators, and then finally, how do you commercialize this in a viable commercial fashion? Another advantage of this is that once we have an idea that's important, such as what we're going to talk about in just a second, about finding new and novel biomarkers in the serum, is how do you synergize with other specialists who have maybe disparate expertise from you, right? So as a clinician or a scientist, perhaps having someone business expertise or legal expertise early on in the process, so that understanding when we come and commercialize this or bring it up to the translation standpoint, how do you merge efforts in a productive and a synergistic fashion? So the big picture summary of this is atherogression in PAD is likely different than other disease processes, and that traditional lipid biomarkers are not reliable for the primary screen, and that people in this room, surgeons, physicians, scientists, should be able to leverage existing biobanks and trials for specimens, including serum and plasma, and that we need synergistic efforts to be able to scale these opportunities and translate them to clinical practice. So these are the fundamental areas of development that we can think about as a group when we come and have Q&A and group discussions. And I want to just give you an example of one way that we are approaching it. It's not the only way, but certainly something that I wanted to include as a topic for inciting discussion. So I've had the great fortune of working with really talented individuals at WashU, and one of the individuals that I've worked with is Guy De Silva, who was a general surgery resident in our program, ultimately did a fellowship and then graduated and now is working in Cleveland Clinic. But through serendipity, Guyan discovered in the serum of patients with CLTI this enzyme that was circulating at really high levels, and it's called fatty acid synthase. And so for the biologists in the room, you can really tell from the name what it does. It produces fatty acids. It's an enzyme that you can turn on and turn off. And it's been known for decades. This enzyme exists in cells in very high proportions in the liver. But we were the first to find it in the serum, and in the serum of people with CLTI. And the way that Guyan found this is that he did traditional experiments that Michael Brown and Goldstein did in 1985, where he took serum and put them through an FPLC column, which is a fast protein liquid chromatography column, and took the different fractions of LDL, VLDL, and HDL, and looked at where does FAS lie. And very few times in science do you find these types of experiments being reproduced. But if you look at this picture right there, that's an FPLC column in the background. This is what Guyan found. He found that 99% of the FAS in the serum is actually in the LDL fraction. And we did immunoprecipitation experiments, and we found that FAS is covalently linked to ApoB, the predominant protein in lipoproteins. Not only that, we did electron microscopy, and when you gold label an antibody for FAS and look at LDL particles, you find FAS sitting on LDL particles. Notice, not all LDL particles have FAS on them. Some of them do, some of them don't. So there seems to be a fraction level, right? For example, there may be a patient with high LDL and low FAS, and vice versa, high FAS and low LDL. And then we went back and we evaluated this using a tissue biobank that we developed at WashU. We have now over 9,000 specimens in this biobank of both tissue, serum, and plasma. So this is a patient with carotid disease. You can see that there's areas of maximal disease and minimal disease in the carotid that we typically as surgeons do all the time, right? And typically we either throw this plaque away or send it off to pathology. But my argument is we should all be collecting this plaque because it could be very vital for our tissue studies and tissue profiling. And what we found was in patients that have carotid disease, areas of maximal plaque had more FAS than areas of minimal disease. So the question is, okay, well, does does this correlate with the serum? And sure enough, we found that in a cohort of patients, people that have high FAS in their serum also had high FAS in their maximal disease tissue. So it seems to be very correlative. And interestingly, patients that have high risk of disease or atherogression, such as patients with diabetes, have higher saturated fatty acids, which are the building blocks for atherogression or vulnerable plaque that are produced by FAS. So our working model is, through additional studies, is that the liver makes these lipoproteins, but these lipoproteins have FAS in them, and FAS is then deposited in the peripheral arterial system. It's a functional enzyme. It makes saturated fatty acids. These saturated fatty acids then contribute to the atherogression. Another resident in my lab, Shirley Tay, came to the lab and did a painstaking study where she collected tibial specimens from amputated legs that we harvested into the biobank. And then she correlated painstakingly ApoB correlation with FAS in a proteomic fashion. And she found that with high certainty that the ApoB in the vessel subendothelium highly correlated with FAS, meaning that they were residing in the subendothelium in the same place, and that this was higher in patients with diabetes. She also repeated what Gayan had done, but in the setting of CLTI. And she found again that people that had high FAS in their tissue also had high FAS in their serum, and it correlated with severity of disease, particularly in CLTI Rutherford status. When she looked at FAS just alone in the serum, FAS alone was an independent predictor of CLTI severity than smoking, with an 85% sensitivity and specificity. Now remember, the cohort studies that I showed you before, the Framingham studies, the Offspring studies, the Speedwell studies, none of them had an odds ratio that surpassed the confidence interval. And now I'm showing you data in a small subset, relatively small subset of 186 patients, that we can detect CLTI severity with 85% sensitivity. That means there's still a false negative rate of 15%. So we could potentially be telling patients that they don't have PAD when they actually do have it by 15%. So there is still room to improve, but it's much better than what we have currently with LDL. Another resident came to my lab and decided, okay, well, let's really investigate this on a bigger scale. Let's look at more patients. So he tapped in our biobank as well and looked at serum specimens for 434 patients. And he did something really interesting. He looked at the traditional lipoproteins, including total cholesterol, HDL, triglycerides, LDL. And I'm just showing you the LDL here. Interestingly, the healthy patients had slightly higher LDL than the patients with asymptomatic PAD and CLTI. And the reason for that is that over 95% of the patients that had CLTI or PAD, luckily, were on statins, right? So the statins are doing their job. They are controlling the LDL. Now, interestingly, he found that the healthy patients had far less FAS compared to their counterparts with asymptomatic PAD or CLTI. And again, we did correlation assessments to see how diagnostic this was. And in this cohort of patients, it was more than 90% sensitive for incidence of CLTI. So the conventional wisdom is that LDL is the truck that sends the fats or the lipids to the artery, causing higher disease. And that more trucks lead higher disease, higher PAD, less trucks lead to less PAD. I'm arguing that it's rather the cargo on the trucks. Less cargo, less disease, higher cargo, more disease. And then we tried to prove this further by knocking it out or giving it inhibitors. And as you can see in rodent studies, whenever we knock out this gene or give it inhibitors, it eliminates plaque. And this eliminates foam cell creation. So I think we have an interesting target. It may not be the end of all, be of all, but it certainly gives us room to think and ponder, is there a better serum biomarker out there that we can use and validate using multidisciplinary efforts to be able to help in the primary screening of PAD? We patented this and are in the process of commercializing it. I started a company on this and have received seed funding to try to be able to develop a kit for CFAS, and this is something that I'd be happy to discuss more with any of you further. Thank you.
It's fantastic. And what I might do is actually, so we went from, precision to personalization to biomarkers and some important biologic questions. I'm going to ask our panel who spoke this morning. So I'm going to ask all of you all to come up to the table for questions. So if I can get, obviously, Katie Wright, our patient representative, Dianna, Milewicz, Mohamed, you're up here. I'll also ask Santhi to come up, Kate and Dawn, all of you all to come up and have a seat. Have a seat right here, you're fine. And we can ask if there are questions for people. I think we have 6 people, so everybody should fit. And once everybody's up here, we have a microphone for you to ask questions in the audience. There are lots of themes. We want to really try to make sure we get across the spectrum of this amazing group of people that have given us these perspectives in quite a wonderful, thoughtful way this morning. Thank you. Anybody so I'll just go over a few notes I took on the questions I heard, and then I'll see if there are people in the audience and stuff. So this morning we heard from, all the signs of getting to patients, and Katie started us off with her story. Then we went to aortic dissections and all of this genetic variation. Mohamed was going after that, but came at the end, which was probably good because we had biomarkers at the end to tell us a little bit about questioning what we think of our own biology, or at least thinking about the spectrum. Santhi gave us a little refresher on making sure we think about common and uncommon variants and how to think about that across hypertension. EHR, AI learning, and then into very rare pediatric groups. So I'll start with a question for the audience while you guys are getting warmed up, but then I'll ask it. And I guess I'll start with our group, and it's just because Mohamed asked this question now. How do you all feel about the basic biologic question that we all walk around with in questioning? Is it a variation of the same disorder, or is atherosclerosis in these different beds, PAD, CAD, cerebrovascular disorder, how different of a disease process is it? I'll ask you, maybe Dianna, you've thought about smooth muscle cells, and you're starting to question, at least in Moya Moya and other things where we're finding variants. What do you think about the concept of that?
The question is whether or not the different beds have different.
Yeah, is the biology, he just identified that, at least he's saying, look, LDL is not the driver of PAD that we think it is. So even though the vascular obviously is disease in all these beds and it's atherothrombosis eventually, but the drivers might be different.
Yeah, and so just talking about smooth muscle cells, when you go along the vascular beds, the origin of smooth muscle cells are different, with the ascending aorta coming from the narrow crest and the descending aorta coming from the mesodermal cells. And so that might impact in terms of the smooth muscle cells migrating into the lesion. But I am struck by the fact with Moya Moya disease and the mid-aortic syndrome, that's the same set of genes. So that kind of does away with that because you're dealing with narrow crest versus mesenchymal-derived smooth muscle cells. And just to mention, I focused on coronary artery disease with our ACTA2 patients, but they also get carotid and peripheral arterial disease, and aortic atherosclerosis is often noted on imaging. So they get it in every single vascular bed, so that doesn't explain that. So I think there are variants that will drive these diseases in multiple different beds, and then there may be specific factors like the FAS that drive it in one bed predominantly. And that leads to one of my questions.
Yeah.
What do you think FAS is doing?
Yeah.
Yeah, it's a great question. So, to answer the first question first, and I'll definitely answer your question as well. So I definitely think that there's more to it than just simple lipoproteins circulating in your plasma. I think that there are certain vascular beds that are more prone to injury, as Dr. Coleman mentioned. There's a reason why you're having stenosis and coarctations in specific areas of the aorta and the renal arteries, and that areas of prior injury are more likely to develop subsequent disease patterns. I think it's the same, I think, in the setting of atheroprogressions. We know, as clinicians and as surgeons, there are vascular beds that are more prone to disease and more prone to more recalcitrant disease that it's much harder to treat. So I think that understanding why these beds are more prone to that disease pattern is very important and may have a lot of the clues. In terms of FAS and how it's working, we did a really cool experiment recently where we took high LDL serum that had little FAS in it, and then we took the opposite, serum that had very high FAS but little LDL, and we added it to macrophages in vitro. And we asked the question, which one would show more foam cells? Now, typically you would say the one that has more LDL would show more foam cells. And we actually saw the opposite. We saw that the one that had less LDL with high FAS had more foam cells. So our hypothesis right now is that FAS enters into the macrophages and causes them to become more foam cell phenotype, leading to the atherogression through the formation of saturated fatty acids like palmitate.
And why is it so high with peripheral vascular disease? Is it smoking or?
Yeah, so I mean, conditions like diabetes are known to increase the expression of FAS in the liver. This has been known for well over 3 decades. In fact, one way to make a rodent diabetic is you overexpress FAS in the liver. And so and they become hyperglycemic, their lipid metabolism becomes altered, the hormones for obesity go up. And when you conditionally knock out FAS in the liver, actually you reverse a lot of these conditions. So you actually decrease the risk of weight gain, you decrease the risk of insulin resistance. So there's a biological imprintation for FAS. Now, if you knock out FAS, for example, in a rodent during embryogenesis, it's lethal. So it's an essential enzyme. You need it to be able to make fatty acids. We need it to make our de novo fatty acids. But when it goes haywire, so to speak, in adulthood, and FAS is not regulated or reined in, it can cause conditions like diabetes or metabolic syndrome.
And those fatty acid, I'm sorry.
Okay, one more.
The foam cells can be either the fatty acids or cholesterol. So are they forming cholesterol foam cells or fatty acid foam cells?
Yeah, so the experiment to be able to know this is you need to conditionally knock out FAS in macrophages. But there's a paper that recently was published by my mentor in Nature where they conditionally knocked out FAS in macrophages. And what they found is that what happens in these macrophages is that they're unable to transport cholesterol. They're unable to absorb
so it
LDL.
does disrupt the
Exactly.
cholesterol
So it
going in.
disrupts it on a membrane level. A lot of cancers, by the way, express FAS to a high degree because they got to make more cell membranes and they got to proliferate and they got to support these cells. And so as a result, FAS is usually upregulated. But if you downregulate FAS, you actually decrease cholesterol transportation as well.
I know we have a question in the audience. We have a few topics in a broad range. Go ahead.
Yeah, just a quick one for Dr. Zayed again. So John Ward with Novartis, you had mentioned that FAS likes ApoB lipoproteins, and so another ApoB lipoprotein is LP(a) or lipoprotein A. Curious if you guys did any work or look there to see if FAS likes LP(a) better, or because there is a correlation between LP(a) and PAD as well as stroke, and just curious any work in that zone.
Yeah, we haven't done that specific substratification of the lipoproteins, lipoprotein A versus B. We do know that it binds covalently to ApoB, and we're actually not the first to discover this. There was actually a proteomic screen that was done well over a decade ago. It pulled down ApoB and it looked at what's stuck to it. And there's a lot of proteins that are stuck to it. FAS did show up on that screen, so it was fun for us to replicate that work and show, indeed, FAS is in the serum, it's catalytically active, and it's bound to ApoB, which makes it even more exciting because it then gives us that model of the truck versus the cargo, right? If it has a way for it to be transported, and it's not just a junk protein in the serum. By the way, FAS is a 272 kilodalton protein, so it's pretty large. Albumin, as a reference, is 500 kilodalton, so it's not a small protein. So for it to be bound makes sense, and for it to be transported and endocytosed also makes sense. So, but we're in the process of trying to correlate FAS to other lipoproteins, like lipoprotein A, lipoprotein B, but also, importantly, CRP, HSCRP, and other imaging studies like coronary calcium stores. And so we're trying to get in touch with prior studies, right, that have done this sort of work and be able to tap in their reserves and see if it correlates with their findings.
Yeah, I'll just put it into the broad thing and then get us to one of the genetic questions that I had, but I'll see if people in the audience had. there's a lot we don't know about the lipid biology that we thought we knew across different things. And clinically, we've been seeing this, right? In the cath lab, I'll say there are patients that have huge burden of peripheral artery disease and have no atherosclerosis in their coronaries. See that frequently and go, how's that possible? Or people that have had a stroke with no atherosclerosis in other beds. And you could just do it phenotypically by saying, who are people that have disease in all 3 beds? Who are people who have disease in just 1 bed and not the other 2 beds? And why is that? And obviously, you're using biology. We could just do phenotypic 2x2 squares all day and say, what are those things? Any other questions? There's another question around earlier that Matt asked, which I'll ask the panel and maybe start with Katie, which was the burden. So we went from genetic testing to the biology down to the plaque, but one of the things that came up was a question about, are we going to get to the place where we do more genetic testing sooner in our patients? And one of the things that we might see are certain things that trigger us to do that. Dianna showed us, well, you might have a facial recognition program that says you might need to think about this patient. As vascular surgeons, you might have a lot of patients you're seeing with aneurysms, a variety of things that you have to decide who needs genetic testing. So where are we, I guess I'll ask across the group. I can certainly talk about our institution when we get to it on genetic testing and what the resources are. But Katie, I'll start with you as someone who's thought probably about this from the patient perspective, and you sort of gave an answer. And you'd even told us in your story you saw a genetic counselor who you wish had more information. I was actually surprised you got to see that genetic counselor. You know, there are times when somebody in your story would say, even though it was on my path report, nobody ever sent me to a genetic counselor, which I suspect you see when you talk to people on the podcast or others. What are your thoughts there?
So just to be clear, the question is my thoughts on where
early genetic testing and how do we get to it, yeah.
Yeah, so I think that early genetic testing is really important, and as you highlighted in your talk, the affordability of genetic testing is, really high right now.
Yeah, the costs are low.
Yeah, the costs are low, the affordability is high. And I think it was Matt that brought up earlier, should we be switching the genetic testing before surgery versus after? And I really liked that question. I think that there's a role for earlier genetic testing. I think there's a lot of things that can be done earlier on if you know what you're dealing with and you're not going in blind. And I'm fascinated by that question, really,
Yeah,
because I think it's important.
It is extremely important, and the information, how to get all that information distilled into something people can understand is going to be a big challenge in genetic counselors. So the test might not be expensive, but I'll tell you, genetic counselors and having enough genetic counselors for people becomes one of the rate-limiting steps at institutions. I know that happens at ours, where we're constantly arguing for more genetic counselors because we can order a genetic test, but we want somebody to help with that.
So just a note, the John Ritter Foundation for many years has funded my lab to provide genetic counseling to whoever contacts them about after they've had a dissection, aortic dissection, or lost a loved one to dissection, just to make sure that people get the information that that dissection can be a red flag that there's other family members at risk. And the treatment guidelines say image first-degree relatives and do genetic testing if there's a familial case. But we can't charge for that because of the regulatory rules across states. So we do it at no cost. So keep that in mind for your patients. If they're anxious and have questions, contact us and they'll get free genetic counseling.
Okay, we're starting to warm up. I got one more statement from Katie, and then I'm going to come down my panel, and then I have some questions.
Well, I just want to add, I never saw a genetic counselor.
Right, sorry.
So I saw a geneticist and then had to kind of recontact that geneticist and was given genetic testing without a genetic counselor. So when this question
a geneticist provides the genetic counseling in that case.
Okay. But I mean, we were talking earlier about, how that impacts counseling pre-genetic testing and how it impacts knowing the impacts on your life before you get tested, life insurance, disability, you're not eligible for those things. And that was information I didn't get before being tested. So I think it's a really important thing to consider as we're kind of bringing this all in.
For sure. Santhi, you had a statement, and then I know.
I was just going to make a comment that I think, the early diagnosis is so critical in knowing, who's at risk. The counter to that is something you said, which is if somebody comes back with, let's say, a COL3A1 variant that we don't know in the VUS category, right? It's it could be a problem, it could be not a problem.
Yeah.
If that ends up in the chart, how does it actually impact care? And we've seen things around that. So just to put that word of caution there about the interpretation and how the information's used.
Yeah, I was just going to say that's one of the things we worry about the most. There's some very clear ones, but there's a lot of gray area that don't. Matt, to you, and then I think Dawn had a question too. So,
Dr. Milewicz, I thought your facial recognition stuff was fascinating. I'm going to show some video data in my talk where we're blurring the faces because the IRB justifiably, I think, says that that's every bit as identifiable as a fingerprint.
Absolutely.
And I wonder, can you comment on how you got that part of your study going, and should we treat a patient's facial image as a protected piece of information, just like we contend with all the ethical issues around genetic testing? Because you could imagine if my facial image is linked to my diagnosis, someone can take like my driver's license photo or even take a photo of me off the web without my permission and learn about me by linking that information. I don't know the answer to that, but I just wanted to say, do you regard that as the same level of identifiable at-risk PHI as genetic information?
So just to answer your first question, when we set up the aortic consortium, we asked for facial pictures and profiles from everybody. And now we're just getting around to using that data now that machine learning has become more prominent. And I agree with you, and we're now getting some pushback from some of the patient groups about doing this. But I think it's always a balance for how much you learn, how many lives you can save with that information and correctly manage them. I mean, all it would have been, we could have taken a picture of Katie and made a diagnosis on that first trip in the ER and prevented a lot of years of misdiagnosis and even, potentially death if she wasn't correctly treated for that splenic rupture.
Yeah.
But I still, it still does better than I do with my eye because there's different types of COL3A1 variants that cause VEDS, and some are very mild, and it still picks up the facial features in those patients, and I can't see it. So I think there still is a role for this. And educating general physicians in dysmorphology, forget it. It's not going to happen.
Yeah. Sorry. Back and forth. Dr. Conti's going to.
I just wanted to sort of reflect on a lot of what we'd heard about this morning were actually genetics, and we're starting to hear more now about some other layers like biomarkers. We started out talking about how we're putting patients into groups, but I want to sort of broaden it about not hardwired things, but things that change over time. So clearly, if you can identify someone who's got a fixed genetic defect and they're going to be in a certain subgroup of patients with a risk for life, that's important. But there's many other things that are potentially modifiable, or maybe we should be thinking more about classifying patients by their trajectory rather than their fixed risk over life. And so how do we fold that together with what we're trying to do, which is also provide ways for patients and providers to maybe alter that, if you will, destiny. So I think I'm sort of asking the broader question about, again, getting back to the original thing about precision medicine, is how we group people. We've got some groups that are very extreme and very fixed phenotypes, but then we have a lot of more folks that are really more characterized maybe by differences in trajectory over time, and even some of these biomarkers obviously are not fixed. You measure them at one time, they may be different at another time. What's the approach that we should take about how we're going to start to bring that element together?
Can I take a step?
Yeah, go ahead.
So one of the things that machine learning can really help us with is in looking at all of that heterogeneity and looking at it over time. And so, for example, somebody may have a biomarker that predisposes them to having more aggressive atherosclerosis, And that person may also have diabetes that's controlled or is not controlled. And that person's diabetes may be controlled or uncontrolled over time. And they're, I don't know what the heck else would influence FAS, but something that they eat may change their cholesterol profile over time as well, right? And so thinking about each of these things individually and how they may affect somebody is impossible to do, with like a priori hypotheses. And so using, it's basically like fancy survival analysis where you identify things that you think could be important over time. For example, wavering A1C levels, wavering LP(a) levels, and you fix those with marks that correlate to other kind of dramatic things in that person's life, right? Like a bypass surgery or a reintervention or an MI. And you can start to draw correlations here. And so that's on the list. We should do that.
Yeah, I would say that your question strikes the heart of the morning to maybe the afternoon, because the morning's been a lot about precision, some about biomarkers, but personalizing behavior and response, that's way more than what we've been talking about and probably has a much bigger impact on the majority of people. And so a conversation we haven't had yet today is, if you think about vascular health, most of it's not in the hospital, most of it's not in the HR. Most of it's on my iPhone, and it's captured by stuff that I surf. So I suspect there's things that we could be thinking about affecting our behaviors of our patients that a lot of people, interestingly, are. And just to what you said, if you knew that, oh, I'm kind of off kilter here with my sugar or my blood pressure, and a little nudge or a little reward or an AI assistant to move the car back into the lane, as I said last night, something to help both clinicians and patients to get them there.
I would say too, just at a very practical level, like we shouldn't treat everybody the same, right? Some of our patients are going to be Johnny on the spot with every single thing that we tell them, and others are not. And there's a million reasons why those barriers may exist. But if we identify a patient who has a lot of barriers, who's struggling, we should treat them differently than those who don't have those same struggles. And whether that's with fluctuating A1Cs or with just a genetic predisposition to having aggressive disease, we can start separating those people out. And one of the other great things about machine learning is that it learns based on response to original treatments. And so for non-responders, it can suggest to you what the next best thing is. And that's something that I think we have a lot to explore in as well.
Yeah, just one last, this is a fascinating discussion. We're talking about a lot of data. A lot of it, as Dr. Conte said, there's value in the longitudinal data of, are things going up, down over time. There's a question for Katie. You as the ultimate stakeholder, for the condition that you have, how do you manage, this is more of a health informatics question, how do you manage all that data? Do you, you're very, I know University of Washington's fantastic, but what if you move to Texas or somewhere else? How do you manage bringing it with you? Do you have it on you at all times? Do you not? What do you think of data interoperability and such?
That is such a great question. So I have moved across the country 3 times between Florida and Washington State and have had to do this. It's not fun. It isn't easy. I think I'm a big user of my chart now, and that has made it a lot easier. And I don't ever want to move again, because it's not, there was, like apps come and go that offer you this, it's like bring your data anywhere and make it really accessible to emergency rooms, and then they disappear. And so you put all of this work into an app, and then it goes away. I think it's a really important thing to consider is how we do that, and I think there's a lot of barriers to getting that data, depending on which provider you're seeing and where you're seeing that provider, and do you have to go and sign a release form to get your own data from them? One of the biggest things that I see with VEDS in particular is that my awesome vascular surgeon, Dr. Shireen Shalhoub, really likes to see the images. So when I was living in Florida and I was continuing to see her in Washington on a periodic basis, it was really difficult to get the actual images sent to her from my provider in Florida. And I think there needs to be, for me, I think that's the biggest need, is for someone with VEDS in the electronic health record is being able to easily get the images themselves, not just the radiology report.
Yeah, talk about something we deal with every day and so real, right? And in whichever part of vascular medicine you work in, wherever you are in whichever cardiovascular imaging, you'd love to have it real time when you're taking care of patients. And it's a struggle. It's getting better, but it's still a struggle that I think really matters. Other questions? I saw, Mark, I see your hands up, and I was interested in your thoughts on some of the conversation, given your... Sorry, yeah.
So I was just going to ask a little bit about the genetic testing and the need for genetic counselors, and whether we need to sort of demystify genetics and package it in a way that tells the person and the patient at the time what they need to know. I mean, I'll give you an example. I ran the aortic center at the Brigham, and now at University of Colorado, we had a patient with a type B dissection extensive who had VEDS and wasn't known in his 50s and had a lot of adverse remodeling. It was big tear, entry tear, lesser curvature, and the procedural team said, you know, we should probably put in an endograft before this patient leaves. Wasn't known that had VEDS. And then when he actually took a reasonable history, had muscle tears and other issues, and so we tested and had a mutation, and we didn't need to know everything about that diagnosis or whether, you know, was it safe to do colonoscopy in the long term. At that point, it was all that we needed to know was it was safe to put an endograft in, which was no, and we're going to choose a different strategy. And I wonder whether, because I do worry that the complexity of understanding all of the implications then leads to fear for some clinicians, so they don't even check, and they say, well, we're going to send them to genetics, and then that's going to be 6 months, and maybe miss an opportunity to drive care. And so I'm just wondering if there's a way to kind of package it for the person who's ordering it for what they need to know.
Yeah, that's why we want to have this system where you can put the gene, the variant in, and it tells you the management A, B, C, D. And then if you say, okay, if it says avoid endovascular repair, it'll pop up all the data that you need to see in terms of poor outcomes with endovascular repair in VEDS patients. You know, the references and sometimes the charts, like, you know, in terms of age of onset of aortic events and stuff, like whatever is relevant to that data.
Yeah, I think, wondering whether you could phenotype the provider who's asking, you know, in a way, so that you don't give them everything, just sort of move it to the top. Here's your issue right now. Do you do a procedure? And then, yeah, there's this other stuff versus packaging it the same way for everyone.
I think that's a great question and a great thought, because I'm sure some of the stuff Don showed us, you know, there's some conditions where there's clear, very rare scenario, but there are others where you're in a, you know, what we don't have in cardiology, I'll say, I don't know about all of vascular medicine, but all of cardiology is going through this. I'll use a non-vascular example. If you show up with non-ischemic cardiomyopathy today in the United States, there's a variety of genetic known disorders that cause you to have an abnormal heart without having a heart attack. Depending on your age, we could argue you should be getting pretty broad genetic testing. Mm-hmm. But we as a group of clinicians and societies haven't come forward and said very clearly, with these sets of pre-test probabilities or scenarios, you should do genetic testing or not. Now, I just want to be very clear. The example I gave you is going to give you a bunch of stuff we're not sure what to do with. I get it. There's going to be some genes you might find where you go, this is very clearly going to progress and we need to have this person in a certain scenario. So I think it's going to be, each group is going to have to get close to saying, in this condition, when you see these things, consider a genetic test, and then this will help you do it. Dawn?
And I do want to add on that. I've got a few thoughts that I just wanted to share and see if it triggers some other concepts. I know Shireen really well. She's a fabulous surgeon. She's super committed to her patients. There are some nuances around the way she offers care and what she offers her patients that's really special. And in this particular space, and in the VED space, it's not entirely dissimilar to how the pediatric renovascular hypertension space works. You've got a few pockets where patients can get super subspecialized care, and they're really reliant on a primary individual or a very small team to be really available to them around the clock. When you listen to Shireen speak, everybody should be genotyped, and that genotype should prompt individualized care, but she acknowledges the limitations in genetic counselors and genetic physicians across the country, so she does it herself. not all vascular surgeons or vascular medicine physicians or cardiologists or other specialists are going to have the time and bandwidth and capabilities to do that. And so as I've continued to think about these patients, and we're working through our own PCORI funding mechanisms right now to optimize access to care and empower our patients to be almost stewards for the direction of their own health, and it gets to what Dr. Conti was referencing. As we build out tools and guidance, we will do our best to create risk predictors. And I think for precision medicine across lots of rare and common diagnoses, that's so contingent on your genotype, but it does consider all of these other factors. You have a tool, but then you have to implement it. And the fact is we've got so much data in our healthcare system, and you've got overburdened physicians. Translating it in a meaningful way to your stakeholders and your patients is going to take an entirely separate set of implementation factors, I think, that have been conceived. And so whatever work comes from this forum, I think it's super important that patients are like aligned elbow to elbow with the work. And I think giving them guidance also that is educated to physicians and helping them be self-advocates, because whether it's a rare disease and having to find a pocket of treatment at Boston Children's or Michigan or Duke, or somebody who's got critical limb-threatening ischemia and they just happen to be in a super rural setting, I mean, the concepts are absolutely all the same. And I think what's really interesting is there are larger implementation level studies and things that could be pushed forward from this work that translate to high and low frequency diseases.
I really love that you brought that up. Oh, sorry.
No, go ahead.
Okay. I think that implementation is something I've thought a lot about, because we have, some, as you mentioned, like having published, like if this, then genetic tests, right? We have that for VEDS. We've had that since 2017 with the publication of the International Classification of Ehlers-Danlos Syndromes. It's there. How does that information get filtered down outside of these super special centers into like where most people are probably living or getting their care? And that's something like as a patient that I've experienced a lot of, is the care that you get obviously is going to be very, very different depending on where you go, but it's going to, even the information, like you have to know enough yourself as a patient to know when you are interacting with a physician that does not have the information that you have, and then navigate that very tricky scenario. One additional thing I wanted to mention, I think it popped into my brain with the last, with his question, is that in the last, for the last 3.5 years, I was building what's called the VEDS movement. It's a division of the Marfan Foundation for VEDS, and we've done a lot of work on developing, patient resources to help advocate in emergency settings, but also to advocate when you are going to see a gastroenterologist or when you're going to see a pulmonologist. And my vision for that was that there would be these fact sheets for each provider, so that, and it would be accessible to providers and the patients to download from the website. One of the challenges is how do you get it to, if you're a patient with the information, how do you give it to a provider and have them believe it?
Yeah, it's really hard, isn't it? It's hard to get behavior change. Well, I was talking about people, but the doctors and the system's one of the hardest to get behavior change in, as you're highlighting. It's been a great panel. I'm cognizant that we have one more talk before lunch, and I want to make sure we get to it, so I want to just give you guys all a hand and thank you for coming up.